Hello there. I'm so glad you found your way here tonight. Right now, as you settle in, imagine a world that has been changing for billions of years. A world where every living thing around you, from the smallest bacterium to the largest whale, carries within it the echoes of countless ancestors. A world where the tiniest differences between individuals have shaped the extraordinary diversity of life we see today. This is natural selection, the quiet, patient force that has sculpted life on Earth since the first self-replicating molecules emerged in ancient seas. A process so elegant that it requires no designer, no plan, only the simple truth that some variations survive better than others. Tonight, we journey into the gentle workings of evolution's engine. We will explore over 100 soothing facts about natural selection, from Darwin's voyage on the Beagle to the antibiotic resistance unfolding in hospitals today, from the peacock's extravagant tail to the silent mutations reshaping genomes every moment. There is no test at the end, no rush, no urgency, just the peaceful unfolding of one of science's most profound ideas to carry you softly towards sleep. So let your breathing slow, let your thoughts grow quiet, and allow yourself to drift into the tranquil wonder of how life has shaped itself across deep time. Welcome to Drowsy Discovery. I am glad you found your way here tonight. Perhaps you have had a long day. Perhaps you simply enjoy falling asleep to thoughts about the remarkable processes that have created all living things. Either way, this space is for you. There is something deeply calming about contemplating natural selection. Every adaptation, every survival strategy, every new species that emerges carries within it a story millions of years in the making. You are connected to this story more than you might realize. The same process that shaped the wings of birds and the fins of fish also shaped the unique features of your own body. The atoms that form your cells were once part of ancient organisms, passed down through an unbroken chain of life spanning billions of years. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. So settle in, let the weight of the day drift away, and let us begin with the first of many quiet truths about natural selection. Natural selection is patient beyond measure. It works not in days or years, but across geological ages. It shapes populations one generation at a time, favoring the variations that happen to fit their environment a little better than the rest. And yet, within all that gradual change, there is an ancient creativity, an endless experiment that has produced everything from photosynthesis to human consciousness. Tonight, we will wander through this magnificent process. We will meet the creatures whose adaptations tell the story of survival. We will discover how simple rules give rise to astonishing complexity, and we will uncover truths about evolution that may surprise even the most curious minds. But there is no hurry. Natural selection has been working for nearly 4 billion years. It can wait a little longer while you rest. Let us begin with what makes this process so extraordinary. Natural selection was first described by Charles Darwin and Alfred Russel Wallace. The theory of natural selection is one of the most important ideas in the history of science, and it was arrived at independently by two British naturalists. Charles Darwin had been developing his ideas quietly for years, drawing on observations made during his famous voyage and on decades of careful study at home. Alfred Russel Wallace, working thousands of miles away in the tropical forests of Southeast Asia, reached strikingly similar conclusions through his own fieldwork. In 1858, both men presented their ideas jointly at a meeting of the Linnean Society in London. an event that attracted surprisingly little attention at the time. It was Darwin who went on to publish the comprehensive account, but Wallace's contribution was essential and is increasingly recognized. Together, they described a mechanism by which populations change over time, driven not by any guiding hand, but by the simple fact that some individuals are better suited to their surroundings than others. It was a quiet revolution, born from patient observation rather than dramatic experiment. Darwin developed his theory after a 5-year voyage on HMS Beagle. In 1831, a young Charles Darwin boarded HMS Beagle as the ship's naturalist for what would become a 5-year voyage around the world. The journey took him from the coasts of South America to the remote Galapagos Islands, from the coral reefs of the Pacific to the forests of Australia. Along the way, Darwin collected thousands of specimens and filled notebook after notebook with observations about the plants, animals, and geological formations he encountered. It was the extraordinary variety of life he witnessed, and the subtle differences between closely related species on neighboring islands that planted the seeds of his revolutionary idea. The voyage did not provide instant clarity, however. It was only after years of further study, correspondence, and reflection back in England that the full picture of natural selection began to take shape in his mind. The Beagle voyage gave Darwin the raw material, but it was his patient, meticulous thinking over the following decades that transformed those observations into a theory that would change our understanding of life itself. The Galapagos finches helped Darwin understand how species diverge. The Galapagos Islands, a volcanic archipelago in the Pacific Ocean, are home to a group of small birds that would become central to evolutionary thinking. Darwin collected specimens of these finches during his visit, but did not initially appreciate their significance. It was only after the ornithologist John Good examined the birds back in England and identified them as distinct but closely related species that Darwin began to see the pattern. Each island harbored finches with slightly different characteristics, particularly in the size and shape of their beaks. The differences corresponded to the types of food available on each island, suggesting that the birds had descended from a common ancestor and diverged over time as they adapted to different ecological niches. This realization was a key piece of evidence supporting the idea that species are not fixed, but change in response to their environments. The Galapagos finches have since become one of the most studied examples of evolution in action, a quiet testament to the power of natural selection working across isolated populations. Each finch species had a beak adapted to its specific food source. The beaks of the Galapagos finches are a beautiful example of form-following function. Species that fed on large, hard seeds evolved thick, powerful beaks capable of cracking tough shells. Those that ate insects developed slender, pointed beaks ideal for probing into bark and crevices. One species even learned to use cactus spines as tools to extract grubs from wood, aided by a beak shaped for precision rather than brute force. The variation in beak shape across the finch species mirrors the range of available food sources across the islands, and it demonstrates how natural selection can sculpt a single ancestral form into many specialized shapes over time. Modern research has identified specific genes that control beak size and shape, confirming that these differences have a clear genetic basis. Ongoing fieldwork in the Galapagos continues to document how beak dimensions shift from generation to generation in response to changes in food availability. It is natural selection made visible, written clearly in the beaks of small birds on remote volcanic islands. Wallace independently arrived at the same theory while in Southeast Asia. Alfred Russel Wallace spent 8 years traveling through the Malay Archipelago, collecting tens of thousands of biological specimens and observing the astonishing diversity of life in the tropical forests. It was during a bout of fever on the island of Ternate that the idea of natural selection crystallized in his mind. He wrote a brief essay outlining the theory and sent it to Darwin, who was stunned to find that Wallace had reached the same conclusions he had been developing privately for 2 decades. The essay prompted the joint presentation of their ideas in 1858. Wallace's contribution to evolutionary theory is sometimes overshadowed by Darwin's more famous publication, but his independent discovery underscores just how compelling the evidence for natural selection truly was. 2 minds, working in very different parts of the world and drawing on different sets of observations, converged on the same fundamental insight. The parallel discovery is itself a remarkable story, a quiet reminder that great ideas sometimes emerge when the evidence is simply too strong to ignore. Darwin waited over 20 years before publishing on the origin of species. After returning from the Beagle voyage, Darwin spent more than 2 decades quietly building his case for natural selection. He studied barnacles in exhaustive detail, corresponded with breeders, botanists, and geologists, and amassed an enormous body of supporting evidence. He was well aware that his ideas would challenge deeply held beliefs about the natural world, and he wanted his argument to be as thorough and irrefutable as possible. It was only when Wallace's letter arrived in 1858, presenting essentially the same theory, that Darwin was spurred to finally publish. On the Origin of Species appeared in 1859 and sold out its first print run on the day of publication. The book presented the case for natural selection with such careful reasoning and abundant evidence that it changed the course of biology. Darwin's long delay is often attributed to a combination of caution, perfectionism, and concern about the social and religious implications of his work. Whatever the reason, the result was a book of extraordinary depth, the product of a patient mind that refused to rush its most important contribution to the world. Natural selection requires heritable variation within a population. For natural selection to operate, there must be differences among individuals in a population, and those differences must be at least partly inherited. If every organism in a group were genetically identical, selection would have nothing to work with. It is the variation in traits, whether in size, color, behavior, or resistance to disease, that gives natural selection its raw material. These differences arise from mutations, recombination during sexual reproduction, and other genetic processes. Some of the variation is visible, such as differences in fur color or beak shape, while much of it is hidden at the molecular level. The key requirement is that the variation can be passed from parent to offspring. Without heritability, a trait that improves survival in one generation could not spread through the population over time. This simple requirement is one of the foundational pillars of the theory. It is a quiet truth that the diversity you see in any group of living things is not just interesting to observe. It is the very engine that allows life to adapt and change. It also requires that some variations affect survival or reproduction. Not all differences between individuals matter to natural selection. Many variations are neutral, making no difference to an organism's chances of surviving or reproducing. But some variations do matter. A slightly thicker coat might help one animal survive a harsh winter better than its neighbors. A marginally faster reaction time might allow a prey animal to escape a predator that catches its slower companions. These differences, even when they are very small, can tip the balance between survival and death, between reproducing successfully and leaving no offspring at all. Over many generations, traits that improve survival or reproduction, even by the tiniest margin, tend to become more common in the population. This is the essence of selection. It does not require dramatic advantages, only consistent ones. The process is entirely indifferent to traits that have no effect on fitness. It is a filter that acts quietly on the variations that happen to make a difference, accumulating small improvements over vast stretches of time. Organisms must produce more offspring than can possibly survive. One of the essential conditions for natural selection is that organisms tend to produce far more offspring than the environment can support. A single oak tree may release millions of acorns in its lifetime, yet only a tiny fraction will ever grow into mature trees. A female cod can produce millions of eggs in a single spawning season, but the vast majority will perish before reaching adulthood. This overproduction creates intense competition for limited resources such as food, space, and mates. Not all individuals will survive, and those that do tend to be the ones best adapted to the conditions they face. Darwin drew inspiration for this idea partly from the work of Thomas Malthus, who wrote about the tendency of human populations to grow faster than their food supply. The same principle applies throughout the natural world. The struggle for existence is not always dramatic. Sometimes it is as quiet as one seedling growing a little taller to reach the sunlight while another is left in the shade. But it is this quiet contest that drives the process of selection forward. Those best suited to their environment are more likely to reproduce. In any given environment, some individuals will be better equipped to find food, avoid predators, resist disease, or attract mates than others. These individuals are more likely to survive long enough to reproduce and pass on their genes. This is often summarized as survival of the fittest, though the word fittest here does not mean strongest or fastest. It simply means best suited to the current conditions. A small, inconspicuous moth may be fitter than a large, conspicuous one if its coloration helps it avoid being eaten. A bacterium with a particular enzyme may be fitter than its neighbors if that enzyme allows it to digest an available food source that others cannot use. Fitness is always relative to the environment, and it changes whenever the environment changes. What is advantageous today may be neutral or harmful tomorrow. This is why natural selection never reaches a final destination. It is always responding to the present, quietly favoring whatever works here and now, with no concern for the future. Over time, advantageous traits become more common in a population. When a trait helps an organism survive and reproduce even slightly better than others, the genes responsible for that trait are passed on to more offspring. In the next generation, a greater proportion of individuals will carry those genes. Over many generations, this gradual accumulation continues until the advantageous trait becomes widespread throughout the population. The process is often slow, taking hundreds or thousands of generations to produce a noticeable change, but it is relentless. Each generation acts as a filter, slightly increasing the frequency of beneficial traits and slightly decreasing the frequency of harmful ones. The result over deep time is adaptation, the remarkable fit between organisms and their environments that is visible everywhere in nature. The spots on a leopard, the venom of a snake, the ability of a seed to lie dormant for years until conditions are right, all of these are the products of advantageous traits accumulating over countless generations. It is a patient process, quiet and unhurried, yet it has produced all the extraordinary complexity and beauty of life on Earth. Natural selection does not have a goal or direction. One of the most important things to understand about natural selection is that it has no purpose, no plan, and no endpoint in mind. It does not strive to create better organisms or drive life toward any particular form. It simply favors whatever traits happen to improve survival and reproduction in the present environment. If the environment changes, the direction of selection changes with it. A trait that was once advantageous can become a liability, and traits that were previously neutral may suddenly become crucial. There is no foresight involved. Natural selection cannot anticipate future conditions or prepare organisms for challenges that have not yet arrived. This lack of direction is one of the reasons evolution can sometimes produce what appear to be imperfect designs, structures that work well enough but carry traces of their evolutionary history. The human spine, for example, is a modified version of a structure originally evolved for walking on 4 legs, and it shows. Natural selection is a tinkerer, not an engineer, working only with the materials available and responding only to the demands of the moment. It simply favors whatever works in the current environment. Natural selection is entirely indifferent to elegance, beauty, or long-term planning. It favors whatever helps an organism survive and reproduce right now in the specific environment it currently inhabits. A trait does not need to be perfect. It only needs to be slightly better than the alternatives available at the time. This means that evolution often produces solutions that are good enough rather than optimal. The panda's thumb, for instance, is not a true thumb at all but a modified wrist bone that serves well enough for stripping bamboo leaves. Similarly, the recurrent laryngeal nerve in mammals takes an unnecessarily long detour through the chest before reaching the larynx, a quirk inherited from fish ancestors. These imperfections make sense when you understand that natural selection can only modify what already exists. It cannot start from scratch. It works with the hand it is dealt, improving what it can, one generation at a time, with no concern for what might be ideal. It is a process of constant adjustment, not deliberate design. The peppered moth is a famous example of natural selection in action. The peppered moth of Britain became one of the most celebrated examples in evolutionary biology during the 19th and 20th centuries. Before the Industrial Revolution, the most common form of the moth had pale, speckled wings that blended well against lichen-covered tree bark. This camouflage protected the moths from hungry birds. A darker form of the moth also existed, but was relatively rare, because it stood out against the pale bark and was easily picked off by predators. The peppered moth story illustrates several key principles of natural selection. It shows that variation exists within populations, that the environment determines which variants survive, and that the composition of a population can change significantly within just a few generations. The simplicity and clarity of this example have made it a staple of biology textbooks around the world. It is a quiet story of survival and adaptation, played out on the trunks of trees. The moths themselves were simply living their lives, unaware that they were demonstrating one of the most powerful principles in all of biology. During the Industrial Revolution, dark moths became more common on soot-covered trees. As factories spread across Britain during the Industrial Revolution, they released enormous quantities of soot into the atmosphere. This pollution killed the pale lichens on tree bark and darkened the surfaces where peppered moths rested during the day. Suddenly, the pale moths that had once been so well camouflaged became conspicuous against the blackened bark, and birds picked them off more easily. The dark form of the moth, previously rare, now had the advantage. Its darker coloring blended with the sooty surfaces, making it harder for predators to spot. Over the course of a few decades, the proportion of dark moths in industrialized areas increased dramatically. This shift was documented by collectors who recorded the changing ratios of light and dark forms over time. It was natural selection in action, visible within a human lifetime. The environment had changed, and the population changed with it, not because individual moths transformed, but because those with the more suitable coloring survived and reproduced at higher rates. It was a somber demonstration that the world shapes life as much as life shapes the world. When pollution decreased, lighter moths rebounded in numbers. In the second half of the 20th century, clean air legislation was introduced across Britain and industrial pollution began to decline. As the air quality improved, lichens gradually returned to tree bark, restoring the pale surfaces that had once been the norm. With the environment shifting back, the selection pressure reversed. The dark moths, which had thrived on soot-covered trees, now stood out against the lighter bark and became easier targets for birds. The pale form, once again well camouflaged, began to increase in frequency. Over the course of several decades, the proportion of pale moths rose steadily in formerly polluted areas, mirroring the earlier shift in reverse. This recovery provided compelling evidence that the original change had indeed been driven by natural selection rather than some other cause. The peppered moth story, taken as a whole, is one of the clearest demonstrations that natural selection can shift the characteristics of a population in response to environmental change and can reverse direction when conditions change again. It is a gentle reminder that natural selection is always listening to the environment. Antibiotic resistance is natural selection happening in real time. When antibiotics were first introduced in the mid-20th century, they seemed like miracle drugs capable of defeating bacterial infections that had killed millions. But bacteria reproduce so rapidly, with some species dividing every 20 minutes, that natural selection operates on them at a pace visible within days rather than centuries. In any population of bacteria, a few individuals may carry random mutations that make them slightly resistant to a particular antibiotic. When the drug is applied, it kills the susceptible bacteria but leaves these resistant individuals alive. With the competition eliminated, the resistant bacteria multiply freely, and within a short time the population is dominated by resistant strains. This is natural selection in its most urgent and visible form. The rise of antibiotic-resistant superbugs is one of the most pressing public health challenges of our time. It is a stark reminder that natural selection is not a historical curiosity confined to textbooks. It is an active, ongoing process with immediate consequences for human health. Bacteria with resistance genes survive and pass on those genes. When an antibiotic is introduced into an environment, it creates a powerful selective pressure. Bacteria that lack resistance are killed off, while those carrying resistance genes survive and go on to divide. Because bacteria reproduce asexually, each daughter cell inherits the resistance gene from its parent. The result is a population that is increasingly composed of resistant individuals. But bacteria have an additional trick that accelerates the process. They can transfer genetic material between one another through a mechanism called horizontal gene transfer. This means that a resistance gene can spread not only from parent to offspring, but also sideways, from one bacterium to another, even across different species. This ability to share useful genes makes bacterial populations remarkably adaptable. A resistance gene that arises in one strain can quickly find its way into others, creating widespread resistance in a short time. It is a sobering illustration of how effectively natural selection operates when generation times are short and gene transfer is fluid. The quiet exchange of genes between bacteria can have profound consequences for the world above. This is why doctors urge patients to complete antibiotic courses. When a patient takes an antibiotic, the drug begins killing the most susceptible bacteria first. After a few days, the patient may feel better because the bacterial population has been reduced dramatically. But if the course is stopped early, some bacteria may survive, and these survivors are likely to be the ones with the highest natural resistance to the drug. Given the chance to multiply again, they can rebuild the population, now enriched with resistance genes. This is why doctors stress the importance of completing the full prescribed course, even after symptoms have improved. Finishing the course maximizes the chance of eliminating the entire bacterial population, including the more resistant individuals. The overuse and misuse of antibiotics in medicine and in agriculture has accelerated the evolution of resistant bacteria on a global scale. Natural selection does not care about human intentions. It responds only to the selective pressures present in the environment. Every incomplete course, every unnecessary prescription provides another opportunity for resistant bacteria to gain the upper hand. It is a quiet arms race between human medicine and bacterial evolution. Pesticide resistance in insects follows the same pattern. The same principles that drive antibiotic resistance in bacteria also apply to insects exposed to pesticides. When a new pesticide is introduced, it is highly effective at first, killing the vast majority of the target insect population. But within that population, a small number of individuals may carry genetic variations that give them some degree of resistance. These survivors reproduce, passing the resistance genes to the next generation. Over repeated applications of the same pesticide, the proportion of resistant individuals grows until the chemical is no longer effective. This cycle has been documented in hundreds of insect species around the world. Some mosquito populations, for example, have developed resistance to multiple insecticides simultaneously, complicating efforts to control diseases such as malaria. The pattern is a vivid demonstration of natural selection operating in real time, driven by the intense selective pressure of a chemical designed to kill. Farmers and public health officials must constantly develop new strategies and rotate chemicals to stay ahead of this evolutionary response. It is a reminder that natural selection never stops adapting to whatever challenges the environment presents. Sexual selection is a special form of natural selection. Natural selection is often thought of purely in terms of survival, but Darwin recognized a distinct component that operates through the competition for mates. He called this sexual selection. While ordinary natural selection favors traits that help individuals survive, sexual selection favors traits that increase an individual's chances of attracting or securing a mate, even if those traits offer no survival advantage or are actually costly. The peacock's cumbersome tail, the elk's heavy antlers, and the elaborate songs of many bird species all evolved primarily because they impressed potential partners. Sexual selection can be divided into two main categories. Intersexual selection involves direct competition between members of the same sex, usually males fighting for access to females. Intersexual selection occurs when one sex, typically the female, chooses mates based on particular traits. Both forms can drive the rapid evolution of exaggerated features that seem at odds with practical survival. It is a reminder that in evolution, leaving offspring is what matters, and sometimes the path to reproduction is paved with beauty rather than efficiency. It favors traits that increase mating success rather than survival. Sexual selection can produce traits that are actively harmful to survival. A bright, conspicuous plumage makes a bird more visible to predators as well as to potential mates. Enormous antlers are heavy, energetically expensive to grow, and can become tangled in vegetation. Yet these traits persist because the reproductive advantage they confer outweighs the survival cost. An individual that lives a slightly shorter life but produces many more offspring will contribute more genes to the next generation than one that lives longer but mates less often. This trade-off between survival and reproduction is a central theme in evolutionary biology. It explains why so many animal species exhibit dramatic differences between males and females, a phenomenon known as sexual dimorphism. Males are often larger, more brightly colored, or more elaborately ornamented than females because they are under stronger sexual selection pressure. The traits that emerge can seem extravagant or even absurd, but they persist because they work. In the economy of genes, it is not longevity that counts, but the number of offspring you leave behind. The peacock's elaborate tail evolved through sexual selection. The male peacock's extraordinary tail is one of the most iconic examples of sexual selection in nature. The shimmering, iridescent feathers with their eye-like patterns serve no practical survival purpose. In fact, the tail is heavy, conspicuous, and makes the bird less agile when escaping predators. Yet it persists because peahens consistently prefer males with the most impressive displays. By choosing mates with large, vibrant tails, females pass on both the genes for the tail and the preference for it. Over many generations, this feedback loop has driven the tail to ever more extravagant proportions. Darwin himself was puzzled by the peacock's tail, famously writing that the sight of it made him feel ill because it seemed to contradict survival-based selection. It was only when he fully developed the concept of sexual selection that the tail made sense. The males that display most effectively gain more mating opportunities, and their genes spread through the population. The peacock's tail is a quiet testament to the power of choice in shaping evolution, a beauty born not from the struggle to survive, but from the desire to be chosen. Peahens prefer males with larger, more colorful tails. Research has confirmed what Darwin suspected. Female peafowl are selective when choosing mates and show a clear preference for males with larger, more symmetrical, and more brightly colored tail displays. Studies tracking the mating success of individual males in wild populations have found that males with more eye spots on their tails tend to attract more females. The eye spots are not merely decorative. Their symmetry and vibrancy may serve as honest signals of the male's overall genetic quality and health. A male weakened by parasites or poor nutrition is less able to produce and maintain a flawless display. By choosing males with the finest tails, females may be selecting partners with genes that confer resistance to disease or other advantages that will benefit their offspring. This is sometimes called the good genes hypothesis. Whether or not females are consciously evaluating genetic quality, the outcome is the same. The preference is inherited alongside the trait it selects for, creating a self-reinforcing cycle that has driven the peacock's tail to its current spectacular form. It is a partnership between beauty and choice, refined across countless generations. The Irish elk's massive antlers were shaped by female preference. The Irish elk, which went extinct roughly 10,000 years ago, possessed the largest antlers of any deer species ever known, spanning up to 3.5 meters from tip to tip. These enormous structures were not primarily weapons for fighting, though males did spar during the rutting season. Many biologists believe they evolved largely through sexual selection, driven by the preference of females for males with the most impressive antlers. Larger antlers signaled strength, health, and access to rich food sources, because only a well-nourished male in peak condition could afford the enormous metabolic cost of growing such structures each year. The antlers were shed and regrown annually, requiring vast quantities of calcium, phosphorus, and energy. Some researchers have suggested that the antlers eventually became so large that they contributed to the species' extinction, particularly as forests expanded and the animals struggled to move through dense woodland. Whether or not this is true, the Irish elk remains a striking example of how sexual selection can push a trait to extreme proportions. It is a quiet cautionary tale written in bone and antler. Birdsong complexity often evolves to attract mates. In many bird species, Males with more complex, varied, and well-structured songs are more successful at attracting females. This preference has driven the evolution of remarkably elaborate vocal performances across the avian world. The nightingale, for example, can produce over 1,000 different song phrases, strung together in sequences that can last for minutes. Song complexity may serve as an honest signal of the male's cognitive ability and overall health, because learning and performing an intricate song requires a well-developed brain and a body in good condition. Females that choose males with the most impressive songs may be selecting for genes that produce healthier, more intelligent offspring. In some species, males with larger repertoires defend better territories and provide more food to their young, adding a practical dimension to the preference. The songs themselves are often learned from older males rather than inherited directly, making birdsong one of the clearest examples of cultural transmission in the animal kingdom. Sexual selection has turned the dawn chorus into a competition of extraordinary beauty, all of it shaped by the quiet choices of listening females. Bowerbirds build elaborate structures to impress females. Male bowerbirds of Australia and New Guinea construct some of the most remarkable courtship displays in the animal kingdom. Rather than relying solely on physical ornaments, they build intricate structures called bowers from sticks, grass, and other materials, then decorate them with colorful objects such as flowers, berries, shells, and even pieces of plastic or glass. Each species favors a different style of bower and a particular color palette. The satin bowerbird, for instance, has a strong preference for blue objects and will travel considerable distances to collect them. Females visit multiple bowers and evaluate them carefully before choosing a mate. Studies have shown that females prefer bowers that are well constructed, richly decorated, and symmetrically arranged. The bower itself is not a nest. It serves no practical purpose beyond impressing the female. Males with the most appealing bowers attract more mates and produce more offspring. This is sexual selection expressed through architecture rather than plumage. The bowerbird's dedication to design is a gentle reminder that the drive to be chosen can inspire extraordinary creativity, even in the quiet forests of the Southern Hemisphere. Natural selection can act on behavior as well as physical traits. When we think of natural selection, we often picture physical adaptations such as camouflage, sharp teeth, or streamlined bodies. But behavior is also shaped by selective pressures. An animal that learns to avoid a particular predator or that develops an effective foraging strategy is more likely to survive and reproduce than one that does not. Over time, behavioral tendencies that improve fitness can become encoded in the genes, making them instinctive rather than learned. The tendency of many animals to freeze when they detect a predator, for example, is an inherited behavior that reduces the chance of being noticed. Parental care, mating rituals, migration routes, and territorial displays are all behaviors that have been shaped by natural selection. Some behaviors are remarkably complex, such as the waggle dance of honeybees, which communicates the direction and distance of food sources to other members of the colony. The evolution of behavior is often harder to trace than the evolution of physical traits because behavior does not fossilize, but it is no less important a target of selection. Every instinct you carry is a whisper from generations past. Migration patterns are partly shaped by selective pressures. The seasonal migrations undertaken by birds, fish, mammals, and insects are among the most impressive behaviors in the natural world. These journeys, often spanning thousands of kilometers, are not random. They have been shaped over countless generations by natural selection. Individuals that migrated at the right time and to the right place found better food supplies, safer breeding grounds, or more favorable climates, and they produced more surviving offspring than those that stayed behind or migrated poorly. Over time, the genetic tendencies underlying successful migration patterns became more common in the population. The timing of migration is often linked to environmental cues such as day length, temperature, and food availability, and the ability to detect and respond to these cues has itself been shaped by selection. In some species, the entire migration route is genetically encoded, allowing young animals to navigate vast distances without guidance from experienced adults. The monarch butterfly, for instance, migrates across generations, with no single individual completing the entire round trip. Migration is a behavior written in genes, tested by survival, and refined by the patient hand of natural selection across deep time. Social behaviors like altruism can also be selected for. At first glance, altruism seems to contradict natural selection. Why would an organism sacrifice its own fitness to help another? If evolution favors individuals that maximize their own reproduction, how can selfless behavior persist? The answer lies in the fact that genes, not individuals, are the ultimate units of selection. If a gene causes an organism to help relatives who carry copies of the same gene, then the gene can spread even if the helper pays a personal cost. This is the foundation of kin selection theory. Beyond helping relatives, reciprocal altruism can also evolve between unrelated individuals if the favor is likely to be returned in the future. Social species that live in stable groups are particularly prone to developing cooperative behaviors because the benefits of mutual aid can accumulate over many interactions. Warning calls in bird flocks, shared nursing in some mammals, and cooperative hunting in wolves and orcas are all examples of socially selected behaviors. Altruism, far from defying natural selection, is one of its most elegant products, a strategy that thrives when the currency of survival is shared. Kin selection explains why organisms help relatives. Kin selection is the idea that natural selection can favor behaviors that benefit an organism's relatives, even at a cost to the individual itself. The logic is straightforward. Because relatives share a proportion of their genes, helping a relative survive or reproduce can indirectly promote the spread of the helper's own genes. The closer the relationship, the greater the proportion of shared genes, and the more likely it is that altruistic behavior will be favored by selection. This principle was formally expressed by the biologist W.D. Hamilton in the 1960s through a concept now known as Hamilton's Rule. It states that an altruistic act is favored when the benefit to the recipient, multiplied by their degree of relatedness to the helper, exceeds the cost to the helper. This elegantly simple equation explains a wide range of cooperative behaviors observed in nature, from ground squirrels giving alarm calls that attract predator attention to themselves, to female elephants helping to raise the calves of their sisters. Kin selection reveals that apparent selflessness can be deeply selfish at the level of the gene. Helping relatives can spread shared genes to the next generation. When an animal helps a sibling, a niece, or a nephew, it is indirectly promoting genes that it shares by common descent. A full sibling shares, on average, 50% of your genes. A niece or nephew shares roughly 25%. Even a cousin shares about 12.5%. If your help increases the reproductive success of these relatives by more than the cost to your own reproduction, then the genes behind the helpful behavior will increase in frequency. This is why many cooperative and altruistic behaviors are directed preferentially toward close kin. Birds that help at the nest, for example, are usually assisting their parents to raise younger siblings. Meerkats that stand guard while the rest of the group forages are more likely to be closely related to the individuals they are protecting. The gene's eye view of evolution makes this pattern understandable. From the perspective of the gene, it does not matter which body it inhabits. What matters is that copies of itself are represented in the next generation. Helping relatives is simply another way to achieve that quiet objective. Worker bees sacrifice reproduction to help the queen's offspring. In a honeybee colony, the vast majority of females are workers that never reproduce. Instead, they devote their entire lives to supporting the colony by gathering food, building comb, defending the hive, and caring for the queen's offspring. This extreme form of altruism puzzled biologists for years until kin selection theory provided an explanation. Due to the unusual genetics of social insects, known as haplodiploidy, worker bees share roughly 75% of their genes with their sisters, more than they would share with their own offspring if they reproduced normally. By helping the queen produce more sisters, the workers are actually spreading their genes more effectively than if they bred independently. This genetic quirk makes the sacrifice of personal reproduction a viable evolutionary strategy. The colony functions almost like a single organism, with each worker contributing to the collective success. Not every detail of eusocial insect behavior fits neatly into kin selection theory, and other factors are also involved, but the fundamental insight remains powerful. The quiet devotion of a worker bee is, at the deepest level, a strategy for genetic survival. This makes genetic sense because workers share genes with siblings. The haplodiploidy system found in bees, ants, and wasps creates unusual patterns of genetic relatedness. Males develop from unfertilized eggs and carry only one set of chromosomes, while females develop from fertilized eggs and carry two. This means that sisters share an unusually high proportion of their genes, roughly 75%, compared to the 50% shared by siblings in most other species. Because each worker is more closely related to her sisters than she would be to her own daughters, she can maximize her genetic contribution to the next generation by helping the queen produce more sisters rather than by reproducing herself. This was one of the key insights that made kin selection theory so compelling when it was first applied to social insects. The arithmetic of relatedness explains why eusociality, the extreme form of cooperative living with sterile worker castes, has evolved independently multiple times in the Hymenopteran insects. It is not a coincidence that the most cooperative societies in the insect world share this particular genetic system. The numbers quietly favor cooperation over independence. Reciprocal altruism occurs when unrelated individuals help each other. Not all cooperation in nature involves relatives. Reciprocal altruism describes situations in which unrelated individuals help one another, with the expectation that the favor will be returned at a later time. This strategy can evolve when individuals interact repeatedly and can recognize one another, because a history of past interactions allows them to identify reliable partners and avoid cheaters. The benefit of being helped when you are in need must outweigh the cost of helping when you are not. Game theory, a branch of mathematics dealing with strategic interactions, has been used extensively to model the conditions under which reciprocal altruism can be maintained by natural selection. The tit-for-tat strategy, in which an individual cooperates on the first interaction and then mirrors the partner's previous action, has been shown to be remarkably successful in repeated interactions. Reciprocal altruism requires certain cognitive abilities, including the capacity to remember individuals and past encounters, which limits it to species with relatively complex brains. It is a quiet social contract, held together by memory and mutual dependence, and it has been observed in primates, dolphins, and several other intelligent social species. Vampire bats share blood meals with hungry roostmates. Vampire bats provide one of the clearest examples of reciprocal altruism in the animal kingdom. These small bats feed exclusively on blood, and because they can starve within just a couple of days if they fail to find a meal, the stakes of an unsuccessful night are very high. When a bat returns to the roost with a full stomach, it will often regurgitate a portion of its blood meal and share it with a roostmate that was unable to feed. Detailed studies have shown that bats are more likely to share with individuals who have shared with them in the past, and they tend to withhold food from those who have refused to reciprocate. This conditional sharing requires the ability to recognize individual roostmates and remember past interactions. The bats also groom each other extensively, strengthening the social bonds that underpin the sharing system. Over time, pairs of bats develop stable relationships built on mutual aid. This is not random generosity. It is a carefully calibrated social strategy shaped by natural selection to maximize each individual's chances of surviving through lean times. It is cooperation built on trust, memory, and the quiet certainty that the favor will be returned. Those who share are more likely to receive help when they need it. The reciprocal altruism observed in vampire bats and other species works because sharing creates a social obligation. Individuals that consistently share are recognized as reliable partners, and they benefit from a network of allies willing to help them in return. Those that refuse to share are gradually excluded from the cooperative network, leaving them vulnerable when they encounter hard times themselves. This pattern creates a selective advantage for cooperative behavior. Over generations, genes that promote sharing and reciprocity spread through the population because the individuals that carry them survive at higher rates. The system is self-policing because cheaters, those who accept help but fail to reciprocate, are punished by exclusion. This dynamic has been modeled extensively using evolutionary game theory, which shows that stable cooperation can evolve even in the absence of kinship, provided that interactions are repeated and individuals can track each other's behavior. The result is a community held together by mutual dependence rather than genetic relatedness. It is a quiet evolution of trust, where being helpful is not just a virtue, but a survival strategy refined by millions of years of natural selection. Camouflage evolves to help prey avoid detection. Camouflage is one of the most widespread and visually striking products of natural selection. Prey animals that blend in with their surroundings are less likely to be spotted by predators, and this survival advantage has driven the evolution of astonishing color patterns and body shapes across the animal kingdom. The sandy hues of a desert lizard, the dappled coat of a fawn lying in the forest, and the mottled skin of a flounder resting on the sea floor are all examples of camouflage shaped by selection over countless generations. The mechanism is straightforward. In any population, individuals whose coloring happens to match their background more closely will survive at higher rates, and they will pass the genes responsible for that coloring to their offspring. Over time, the match between the organism and its environment becomes increasingly precise. Some species have evolved the ability to change their coloring actively, adjusting their appearance in real time to match changing backgrounds. Camouflage is a quiet art, perfected by the patient accumulation of small advantages over an immensity of time. Stick insects resemble twigs so closely that predators overlook them. Stick insects are among the most remarkable camouflage artists in the natural world. Their elongated bodies, muted colors, and textured surfaces mimic the twigs and branches of the plants they live on with uncanny accuracy. Some species even sway gently in the breeze to replicate the movement of a real branch. This extraordinary resemblance did not appear overnight. It is the product of millions of years of natural selection, during which individuals that most closely resembled their surroundings survived longer and produced more offspring. The evolutionary pressures are intense because stick insects are relatively slow-moving and have few defensive options beyond concealment. Their camouflage is their primary survival strategy. Some species carry the mimicry further by producing eggs that resemble plant seeds, complete with a fleshy structure called a capitulum that attracts ants. The ants carry the eggs underground, inadvertently protecting them from parasitic wasps. Every detail of the stick insect's form and behavior has been honed by selection to avoid detection. It is a creature designed by evolution to disappear in plain sight, a quiet master of the art of not being seen. Leaf-tailed geckos blend perfectly with tree bark. The leaf-tailed geckos of Madagascar are among the most masterfully camouflaged reptiles on Earth. Their flattened bodies, irregular outlines, and bark-like skin patterns allow them to press against tree trunks and virtually disappear. Some species have fringed skin along their jaws and bodies that eliminates the sharp shadow that would otherwise give away their outline, blending seamlessly into the textured surface of the bark. Their eyes, too, are patterned to match their surroundings, making them almost invisible even when viewed at close range. This camouflage is essential for their survival, as Madagascar is home to numerous predatory birds and snakes that hunt by sight. Any gecko that stands out against the bark is at immediate risk. Over countless generations, natural selection has favored individuals whose appearance most closely matches the specific trees on which they rest. The result is a gecko that looks less like an animal and more like a piece of the tree itself. When night falls, they become active hunters, stalking insects through the forest canopy. But during the day, they are nature's invisible masters, resting in plain sight while the world moves past them. Predators also evolve camouflage to ambush prey more effectively. Camouflage is not exclusively a defensive adaptation. Many predators have evolved coloring and body shapes that conceal them from their prey, allowing them to get close enough to strike before being detected. The ambush strategy depends on the predator being invisible until the last possible moment. Tigers, for example, have striped coats that break up their outline in tall grass, making them difficult to spot despite their enormous size. Crocodiles rest motionless in murky water, their rough, dark skin blending with the riverbed, waiting for an unsuspecting animal to approach. The stonefish, one of the most venomous creatures in the ocean, resembles a rock so convincingly that prey swim right over it without noticing. In each case, natural selection has favored individuals whose appearance most effectively conceals them in their hunting environment. A predator that is spotted too early loses its meal, and over time, this consistent pressure produces increasingly refined camouflage. The evolutionary arms race between predator and prey ensures that both sides are constantly improving. It is a silent contest, played out over millions of years, where invisibility is the greatest weapon. The orchid mantis mimics flower petals to lure insects. The orchid mantis of Southeast Asia is one of the most extraordinary predatory mimics in the natural world. Its limbs are flattened and shaded in tones of pink and white, closely resembling the petals of a flower. When it sits motionless among the blossoms, pollinating insects approach it expecting nectar and instead find themselves seized by a lightning-fast predator. Research has shown that the orchid mantis does not merely match the flowers around it. In some cases, it actually attracts more pollinators than real flowers because its coloring and shape are an exaggerated superstimulus that insects find irresistible. This form of aggressive mimicry is the product of natural selection operating on the predator rather than the prey. Individuals that more closely resembled flowers caught more food and survived to reproduce at higher rates. Over many generations, the resemblance became increasingly refined. The orchid mantis is a vivid demonstration that natural selection shapes predators as relentlessly as it shapes prey. The beauty of a flower can be a deadly illusion, perfected by the patient accumulation of subtle improvements across an immensity of evolutionary time. Mimicry is another product of natural selection. Mimicry occurs when one species evolves to resemble another species or a feature of the environment in a way that provides a selective advantage. It is one of the most fascinating outcomes of natural selection because it demonstrates how thoroughly evolution can reshape an organism's appearance, behavior, or chemistry to exploit the responses of other species. Some harmless animals mimic dangerous ones to deter predators. Some predators mimic harmless organisms to lure prey. Some plants mimic the scent or appearance of female insects to trick males into pollinating them. The range of mimicry strategies in nature is extraordinary, and each one has been refined by the same process of variation, selection, and inheritance. Mimicry evolves gradually. A vague resemblance to a dangerous model may provide a small survival advantage, and natural selection amplifies that resemblance over many generations until the mimic is nearly indistinguishable from the original. The precision of some mimicry systems is so remarkable that early naturalists struggled to believe they could have arisen without deliberate design. But they did, through the quiet, steady work of selection acting on random variation. Harmless species may evolve to resemble dangerous ones. In the natural world, looking dangerous can be just as effective as being dangerous. Many harmless species have evolved to closely resemble toxic, venomous, or otherwise unpleasant animals, gaining protection from predators without having to invest in the costly production of poisons or weapons. This strategy works because predators learn to avoid animals that have caused them pain or illness in the past, and they extend that avoidance to any creature that looks similar. A harmless hoverfly that resembles a wasp benefits from the wasp's fearsome reputation without needing a sting of its own. A non-venomous snake, patterned like a coral snake, deters predators that have learned to avoid the real thing. The resemblance does not need to be perfect. Even a rough similarity can provide some degree of protection, and natural selection gradually refines the likeness over time. The key requirement is that the predators in the area must have encountered the dangerous model and learned to avoid it. If the harmless mimic becomes too common relative to the dangerous model, the protection weakens because predators encounter more harmless individuals and learn to ignore the warning. It is a delicate balance maintained by selection. This is called Bayesian mimicry. Bayesian mimicry is named after the 19th century naturalist Henry Walter Bates, who first described the phenomenon while studying butterflies in the Amazon rainforest. Bates noticed that certain palatable butterfly species closely resembled toxic ones in color and pattern. He reasoned that the resemblance was no coincidence, but an adaptation that protected the harmless species from predators. The toxic species serves as the model, and the harmless species is the mimic. Predators that have learned to avoid the model through unpleasant experience also avoid the mimic, giving it a survival advantage. Bayesian mimicry can only be maintained by natural selection if the mimic remains relatively rare compared to the model. If the mimic becomes too common, predators will encounter it more often than the model, and the association between the warning pattern and unpleasant consequences will weaken. This frequency dependence creates a natural check on the population size of the mimic. Bates's insight was one of the earliest examples of how natural selection could be invoked to explain a complex biological phenomenon. It remains one of the most elegant case studies in evolutionary biology. The viceroy butterfly mimics the toxic monarch butterfly. The viceroy butterfly of North America is one of the most well-known examples of mimicry. For many years, it was considered a textbook case of Bayesian mimicry because it closely resembles the monarch butterfly, which is toxic to many predators due to the chemicals it accumulates from milkweed plants during its caterpillar stage. The viceroy shares the monarch's orange and black wing pattern, and predators that have learned to avoid monarchs also tend to avoid viceroys. However, more recent research has complicated the story. Studies have shown that viceroys are themselves mildly unpalatable, which means the relationship may be closer to Mullerian mimicry, where both species are unpleasant and reinforce each other's warning signal. Regardless of whether the mimicry is purely Batesian or partly Mullerian, the evolutionary principle remains the same. Natural selection has favored viceroys that most closely resemble monarchs because the resemblance deters predators. The more precise the mimicry, the greater the protection. It is a quiet evolutionary partnership shaped by the feeding choices of birds over millions of years. Predators avoid both because they cannot tell the difference. The effectiveness of mimicry depends on the inability of predators to distinguish between the model and the mimic. When a bird has a painful encounter with a toxic monarch butterfly, it learns to associate that particular color pattern with an unpleasant experience. The next time it sees a butterfly with a similar pattern, whether it is another monarch or a viceroy mimic, it avoids it. The predator's decision is based on past experience and visual recognition, not on any deeper knowledge of which species is actually toxic. This inability to tell the difference is what gives the mimic its protection. The more accurately the mimic reproduces the model's appearance, the more effective the deception becomes. Natural selection sharpens this resemblance over time because mimics that are slightly less convincing are more likely to be attacked and killed. The predator's learning ability also plays a role. If a particularly observant predator learns to detect subtle differences between model and mimic, it gains access to a food source that others avoid. This creates a counterpressure on the mimic to improve its disguise even further. The result is an ongoing evolutionary dialogue between predator perception and prey deception. Mullerian mimicry occurs when dangerous species resemble each other. Mullerian mimicry, named after the German naturalist Fritz Müller, describes a situation in which two or more genuinely dangerous or unpalatable species evolve to look alike. Unlike Batesian mimicry, where a harmless species free rides on the reputation of a harmful one, Mullerian mimicry is a cooperative arrangement. Each species benefits because the shared warning pattern reinforces the lesson in the minds of predators. When a young bird has a bad experience with one species carrying the shared pattern, it avoids all similarly marked species, protecting individuals from both populations. The more species that share the pattern, the faster predators learn to avoid it, and the fewer individuals of each species are lost during the predator's learning period. This mutual benefit means that natural selection drives the participating species toward a common appearance over time. Many toxic butterfly species in the tropics share remarkably similar color patterns for exactly this reason. The result is a community of dangerous organisms that, despite being only distantly related, converge on the same warning signal. It is cooperation written in color and pattern, reinforced by the simple arithmetic of shared risk. Multiple toxic species share warning colors to reinforce the message. Across the tropical forests of Central and South America, many unrelated species of poisonous frogs, toxic butterflies, and venomous insects share strikingly similar color combinations of red, yellow, and black. These bold, high-contrast patterns serve as warning signals, known as aposematic coloration, advertising to potential predators that the bearer is dangerous or unpleasant to eat. When multiple toxic species share the same warning colors, predators need fewer individual encounters to learn the lesson. Each species contributes to the education of the predator community, and every individual benefits from the collective reputation of the shared pattern. This sharing reduces the cost of being a training experience for a naive predator, because the losses are spread across many species rather than borne by one. The result is a system in which bold, conspicuous coloring is selectively advantageous, the opposite of camouflage. Natural selection can favor both being visible and being invisible, depending on the circumstances. For toxic species, standing out is the point. The brighter and bolder the signal, the more effectively it communicates danger, and the less likely the predator is to make a costly mistake. Predators learn to avoid the pattern more quickly. The speed at which a predator learns to avoid a warning pattern depends on how frequently it encounters that pattern and how unpleasant the experience is. When several toxic species share the same bold coloration, the predator encounters the warning signal more often than it would if each species had its own unique pattern. This increased encounter rate accelerates learning. A young bird might need only 1 or 2 bad experiences with the shared pattern before it learns to avoid all similarly marked animals. This benefits every species carrying the shared signal because fewer individuals are sacrificed during the predator's education. Models of Mullerian mimicry show that the mathematics strongly favor convergence on a common pattern. Any rare variant that deviates from the established signal loses the protection of the shared reputation and suffers higher predation rates. This frequency-dependent selection keeps the shared pattern stable once it is established. The result is a public education system maintained by natural selection, where the cost of teaching predators is shared across an entire community of unrelated species. It is efficiency at work in the natural world, quiet and effective. Convergent evolution produces similar traits in unrelated species. Convergent evolution is the process by which organisms that are not closely related independently evolve similar features in response to similar environmental challenges. It is one of the most compelling demonstrations of the power of natural selection because it shows that given similar problems, evolution often arrives at similar solutions. The streamlined bodies of dolphins and sharks, the wings of birds and bats, and the camera eyes of vertebrates and octopuses all evolved independently in lineages that diverged hundreds of millions of years ago. In each case, the physical demands of the environment, whether swimming efficiently, flying, or seeing in detail, imposed a narrow range of workable designs, and natural selection converged on those designs from very different starting points. Convergent evolution does not mean that the solutions are identical at the genetic or structural level. A bat wing and a bird wing are built from very different underlying structures, but the overall function is remarkably similar. This pattern reveals that the laws of physics and the demands of ecology constrain what natural selection can produce, channeling the creativity of evolution along predictable paths. Sharks and dolphins both evolved streamlined bodies for swimming. Sharks are fish that have been swimming in the oceans for over 400 million years. Dolphins are mammals that returned to the sea roughly 50 million years ago from land-dwelling ancestors. Despite their vastly different evolutionary histories, both have evolved strikingly similar body shapes: streamlined, torpedo-like forms with dorsal fins, tail structures that generate forward thrust, and smooth skin that reduces drag. This convergence is not a coincidence. The physics of moving efficiently through water favor a narrow range of body shapes, and natural selection has driven both lineages toward the same optimal design. The details differ. A shark's tail moves side to side, while a dolphin's tail moves up and down, reflecting their different ancestral body plans. A shark breathes through gills, while a dolphin must surface to breathe air, but the overall silhouette is remarkably similar. This example of convergent evolution illustrates a fundamental principle. When the environmental challenge is the same, natural selection tends to produce comparable solutions regardless of the lineage. It is the quiet logic of physics, expressed through biology. Wings evolved independently in birds, bats, and insects. Flight has evolved at least 4 separate times in the history of life on Earth: in insects, in pterosaurs, in birds, and in bats. Each group developed wings from entirely different precursor structures. Insect wings are extensions of the exoskeleton. Bird wings are modified forelimbs with feathers. Bat wings are formed from skin stretched between elongated finger bones. Pterosaur wings were supported by a single, enormously elongated finger. Despite these structural differences, all 4 types of wing perform the same basic function, generating lift and enabling powered flight. The fact that flight evolved independently on multiple occasions is powerful evidence that the aerial niche provides such a strong selective advantage that natural selection has found a way to exploit it again and again. Each solution reflects the constraints and opportunities of the ancestral body plan from which it arose. The convergence is in function, not in structure. It is a striking demonstration that evolution is endlessly creative within the limits imposed by physics and anatomy. The sky above has been a destination that life has reached by several different routes. Eyes have evolved independently over 40 times. The eye is often cited as an example of a structure so complex that it could not possibly have evolved by natural selection. In reality, eyes have evolved independently more than 40 times across the animal kingdom, in groups as distantly related as vertebrates, mollusks, arthropods, and jellyfish. This remarkable convergence demonstrates that natural selection is more than capable of producing complex organs when the selective advantage is strong enough. The ability to detect light provides such a significant survival benefit that even the simplest light-sensitive patch is better than nothing. From these humble beginnings, natural selection can gradually refine the structure, adding a depression that improves directional sensitivity, then a small opening that creates a pinhole camera effect, then a lens that focuses light, and so on. Each step offers a measurable advantage over the previous one, and each can be achieved through small genetic changes. The diversity of eye designs found in nature, from the compound eyes of insects to the camera eyes of squid, reflects the many different paths that evolution has taken toward the same functional goal. It is one of nature's most eloquent answers to the question of what natural selection can achieve. This shows that similar environments produce similar solutions. The repeated appearance of similar adaptations in unrelated lineages is one of the strongest pieces of evidence that natural selection, rather than chance, is the driving force behind evolutionary change. If evolution were random, there would be no reason to expect the same solutions to arise independently in different groups. But because natural selection is constrained by the laws of physics, chemistry, and ecology, organisms facing similar challenges tend to evolve similar adaptations. Desert plants on different continents have independently evolved thick, water-storing stems and reduced leaves. Burrowing mammals in different parts of the world have independently evolved compact bodies, powerful forelimbs, and reduced eyesight. Deep-sea fish in unrelated families have independently evolved bioluminescence. Each example reinforces the principle that the environment acts as a filter, consistently selecting for certain solutions over others. Convergent evolution does not mean that the outcomes are inevitable, because history and chance also play a role. But it does mean that the range of effective solutions to any given problem is limited, and natural selection reliably finds its way to those solutions. The patterns are quiet but unmistakable. Divergent evolution produces different traits from a common ancestor. While convergent evolution draws unrelated species toward similar forms, Divergent evolution pushes related species apart. When populations of a single ancestral species become separated or face different environmental conditions, natural selection drives them along different evolutionary paths. Over time, the descendants may become so different in appearance, behavior, and ecology that their shared ancestry is no longer obvious without careful analysis. The classic example is the adaptive radiation of mammals after the extinction of the dinosaurs. From a small, insectivorous ancestor, mammals diversified into whales, bats, elephants, horses, primates, and countless other forms, each adapted to a different way of life. The bones in a whale's flipper, a bat's wing, a horse's leg, and a human hand all share the same underlying skeletal pattern, revealing their common origin. But natural selection has reshaped those bones so dramatically that the resemblance is hidden beneath very different surfaces. Divergent evolution is the process that generates the vast diversity of life from shared beginnings. It is the quiet unfolding of possibility from common roots. The forelimbs of whales, bats, and humans share the same bone structure. If you compare the forelimb of a whale, the wing of a bat, and the arm of a human, the external appearance is strikingly different. A whale's flipper is broad and flat for steering through water. A bat's wing is a thin membrane stretched between long, delicate finger bones. A human arm is built for grasping and manipulating objects. Yet beneath these different surfaces lies the same basic arrangement of bones. A single upper arm bone, two forearm bones, a cluster of wrist bones, and a set of digits. This shared pattern is a hallmark of homology, the presence of similar structures inherited from a common ancestor. The ancestor of all three groups had a forelimb built on this plan, and natural selection has modified it in each lineage to suit its particular way of life. No intelligent designer would use the same blueprint for such different functions. But evolution, which can only modify existing structures, had no choice but to work with the materials available. The result is a family resemblance hidden beneath the surface, a quiet signature of common descent written in bone. These homologous structures reveal common ancestry. Homologous structures are one of the most important lines of evidence for evolution by natural selection. When different species share the same underlying anatomy despite using it for very different purposes, it strongly suggests that they inherited that anatomy from a shared ancestor. The more structures two species share, the more recently they are likely to have diverged. Comparative anatomy was one of the earliest fields to provide support for evolutionary theory, and it remains one of the most persuasive. Biologists have identified homologous structures throughout the animal and plant kingdoms, from the vertebrate limb to the arrangement of bones in the inner ear, from the basic body plan of insects to the structure of flowers in different plant families. Homology is not limited to anatomy. Genes, proteins, and developmental pathways can also be homologous, reflecting shared evolutionary history at the molecular level. The discovery of deep genetic homologies, such as the Hox genes that control body plan development across vastly different animal groups, has reinforced the evidence from anatomy with molecular precision. Homology is the quiet thread connecting the tapestry of life. Adaptive radiation occurs when one species diversifies rapidly. Adaptive radiation is the process by which a single ancestral species gives rise to many descendant species, each adapted to a different ecological niche, often in a relatively short period of geological time. It typically occurs when an organism gains access to a new, resource-rich environment with few competitors. The newly available niches present a range of opportunities, and natural selection drives different populations to specialize in different directions. The result is a rapid burst of speciation and diversification. Classic examples include the cichlid fish of the African Great Lakes, which have diversified into hundreds of species with different diets, body shapes, and coloration within just a few million years. The Hawaiian silver swords, a group of plants descended from a single ancestor, have radiated into forms as varied as compact cushion plants, tall trees, and trailing vines. Adaptive radiation demonstrates the remarkable speed with which natural selection can produce diversity when the opportunity arises. It is evolution in fast forward, driven by the opening of new ecological possibilities. Darwin's finches are a classic example of adaptive radiation. The finches of the Galapagos Islands are among the most famous examples of adaptive radiation in the biological world. All of the Galapagos finch species are believed to have descended from a single ancestral species that arrived on the islands from the South American mainland several million years ago. Once established on the archipelago, the founding population diversified into roughly 14 species, each adapted to a different ecological niche. Some evolved large, crushing beaks for hard seeds. Others developed slender beaks for catching insects. One species even uses tools to extract grubs from wood. This radiation was driven by the availability of unoccupied niches on the isolated islands, where competition from other bird species was minimal. Natural selection shaped each population according to the specific food sources and habitats available on its particular island. Long-term field studies have documented natural selection acting on beak size and shape in real time, as populations respond to shifts in seed availability caused by changes in rainfall. The Galapagos finches remain a living laboratory for the study of evolution, quietly demonstrating the principles Darwin first glimpsed nearly 2 centuries ago. Hawaiian honey creepers diversified to fill many ecological niches. The Hawaiian honey creepers are a spectacular example of adaptive radiation on an isolated island chain. All of the species in this group descended from a single finch-like ancestor that arrived in Hawaii millions of years ago. In the absence of competition from the mainland bird species that occupy similar ecological roles elsewhere, the honey creepers diversified into over 50 known species, each adapted to a different niche. Some evolved long, curved bills for extracting nectar from tubular flowers. Others developed thick, parrot-like bills for cracking seeds or short, pointed bills for catching insects. The diversity of bill shapes across the honey creeper family rivals that of the Galapagos finches and demonstrates the same principles of natural selection acting on isolated populations. Sadly, many honey creeper species have gone extinct since human settlement of the islands, and several more are critically endangered due to habitat loss, introduced predators, and avian diseases carried by introduced mosquitoes. The surviving species remain a vivid illustration of what natural selection can produce when given an empty canvas and the time to fill it. It is a story of both extraordinary creativity and fragile beauty. The Cambrian explosion saw rapid diversification of animal body plans. Roughly 540 million years ago, the fossil record reveals a sudden and dramatic appearance of almost all the major animal body plans that exist today. This event, known as the Cambrian explosion, occurred over a period of roughly 20 to 25 million years, a mere blink in geological terms. Before the Cambrian, most life was relatively simple. Afterward, the oceans were populated by arthropods, mollusks, chordates, echinoderms, and many other groups, some of which have no living descendants. The causes of this rapid diversification remain a subject of active research. Some scientists believe that rising oxygen levels, the evolution of eyes, the development of new genetic regulatory systems, or an evolutionary arms race between predators and prey may all have played a role. Whatever the trigger, the Cambrian explosion demonstrates that natural selection can produce rapid and dramatic change when the conditions are right. The burst of innovation laid the groundwork for all the animal diversity that has followed over the past half-billion years. It is one of the most remarkable chapters in the history of life, a quiet explosion that shaped the world. Natural selection drives speciation when populations become isolated. Speciation, the formation of new species, is one of the most important outcomes of natural selection. It typically begins when a population is divided into two or more groups that can no longer exchange genes. This separation can be caused by a physical barrier, such as a mountain range, a river, or a rising sea level, or it can arise from behavioral or ecological differences within a single habitat. Once isolated, each group is exposed to its own set of environmental conditions and selective pressures. Over time, natural selection drives the accumulation of genetic differences between the groups. If enough differences accumulate, the populations may eventually become so distinct that they can no longer interbreed even if the barrier is removed. At that point, they are considered separate species. This process, known as allopatric speciation, is thought to be the most common mode of species formation. It can take thousands or millions of generations, depending on the strength of the selective pressures and the degree of isolation. Speciation is the quiet engine that has generated the millions of species on Earth today. Geographic isolation allows populations to evolve independently. When a population is split by a physical barrier, the two resulting groups experience different environments, face different predators, and encounter different food sources. Even if the initial differences are subtle, natural selection acts on each group independently, favoring different traits in response to different conditions. Random genetic drift also causes the two groups to diverge, particularly if one or both are small. Over time, the accumulated genetic and phenotypic differences can become substantial. The populations may develop different coloration, different body sizes, different feeding behaviors, or different mating calls. These changes reduce the likelihood that individuals from the two groups would recognize each other as potential mates if they were brought back together. Geographic isolation does not guarantee speciation. If the two populations are reunited before sufficient differences have accumulated, they may merge back into a single interbreeding population. But if the separation lasts long enough, the divergence becomes irreversible. Islands, mountain ranges, glaciers, and expanding deserts have all served as barriers that have driven the formation of new species throughout evolutionary history. Each barrier is a quiet starting point for a new branch on the tree of life. Over time, they may become so different that they cannot interbreed. The ultimate consequence of prolonged isolation and divergent selection is that two populations may accumulate so many genetic differences that they can no longer produce viable offspring together. This is the point at which biologists typically recognize them as separate species. The barriers to interbreeding can take many forms. The populations may breed at different times of year, use different mating signals, or develop physical incompatibilities that prevent successful reproduction. Even if mating does occur, the hybrid offspring may be infertile or poorly adapted to either parent's environment. These reproductive barriers are not always sharp. There are many examples of closely related species that can still hybridize in certain conditions, and the boundary between species is often fuzzier than textbooks suggest. Nevertheless, the general trend is clear. As populations diverge under the influence of natural selection and drift, the probability of successful interbreeding declines. The formation of reproductive isolation marks one of the most important thresholds in evolution. It is the moment when diversity becomes permanent and a single lineage becomes two. The Grand Canyon separates two species of ground squirrel. The Kaibab and Harris's antelope ground squirrels live on opposite sides of the Grand Canyon in the southwestern United States. Despite their close resemblance, they are recognized as distinct species, each confined to its own rim of the canyon. The two populations are thought to have descended from a single ancestral species that was divided when the Colorado River carved the canyon deeper and wider over millions of years. Once separated by the vast chasm, the two groups evolved independently under slightly different environmental conditions. The kaibab squirrel, found on the north rim, has a darker underside and a white tail, while the Harris's antelope squirrel, found more widely to the south, has a lighter belly and a gray tail. These differences, while modest, reflect the independent evolutionary paths each population has taken since their separation. The Grand Canyon squirrels are often used as a textbook example of allopatric speciation in its early stages. Whether the two populations have diverged enough to be fully reproductively isolated remains a topic of study. The canyon between them is a quiet reminder that geography can be as powerful as any predator in shaping the course of evolution. They descended from the same ancestor before the canyon formed. Before the Grand Canyon reached its current depth and width, the ancestral population of ground squirrels likely ranged freely across the region. As the Colorado River cut deeper into the rock over millions of years, it eventually created a barrier too wide and too deep for the small squirrels to cross. The populations on either side were effectively isolated from one another, and gene flow between them ceased. From that point forward, each population was subject to its own local conditions, its own predators, its own climate, and its own random genetic changes. Natural selection acted independently on each group, and drift contributed additional differences. The squirrels did not decide to become different. They simply responded to the quiet, unrelenting pressures of their respective environments. Over time, the accumulated changes made each population recognizably distinct. This is the fundamental mechanism of allopatric speciation, repeated countless times throughout the history of life, wherever geographical barriers have divided once continuous populations. The formation of the Grand Canyon was a geological event. The divergence of the squirrels was its biological consequence. One patient process gave rise to another. Coevolution occurs when species evolve in response to each other. Coevolution is the process by which two or more species reciprocally influence each other's evolution over time. When one species changes, it alters the selective pressures acting on the other, which in turn evolves in response, which changes the pressures on the first species, and so on. This feedback loop can drive rapid and dramatic evolutionary change. Coevolution is especially common in relationships that involve close ecological interactions, such as predation, parasitism, pollination, and competition. A predator that evolves a faster running speed imposes selection on its prey to become faster as well. A parasite that evolves a new way to evade the host's immune system drives the host to evolve a better immune response. Each species acts as part of the selective environment of the other. Coevolution can be antagonistic, as in the arms race between predators and prey, or mutualistic, as in the close partnerships between flowering plants and their pollinators. In both cases, the result is a deepening entanglement, an evolutionary dance in which neither partner can stop moving without falling behind. It is a quiet dialogue between species carried on across millennia. Flowers and their pollinators often co-evolve together. The relationship between flowering plants and their pollinators is one of the most celebrated examples of coevolution in the natural world. Flowers have evolved an extraordinary range of colors, shapes, scents, and nectar rewards to attract specific pollinators, while the pollinators have evolved corresponding adaptations to access the resources the flowers offer. Hummingbirds have long, slender bills that fit perfectly into tubular flowers. Bees can see ultraviolet patterns on petals that guide them to the nectar. Certain flies are attracted to flowers that smell of rotting meat, pollinating them while searching for a place to lay their eggs. Over millions of years, these reciprocal adaptations have become increasingly refined. In some cases, a plant species and its pollinator have become so dependent on each other that neither can survive without the other. This obligate mutualism represents the deepest possible co-evolutionary entanglement. The beauty of a flower is not merely aesthetic. It is functional, shaped by natural selection to attract exactly the right visitor. Each bloom is a quiet invitation, designed over millennia for one particular guest. Some orchids have tubes that match the tongue length of specific moths. One of the most famous predictions in the history of evolutionary biology involves an orchid and a moth. When Darwin examined the star orchid of Madagascar, he noticed that its nectar was stored at the bottom of an extraordinarily long spur, roughly 30 centimeters in length. He predicted that somewhere on the island there must exist a moth with a tongue long enough to reach the nectar, because natural selection would have favored any pollinator capable of accessing so rich a reward. Decades later, that moth was discovered, a hawk moth with a proboscis of the predicted length. This tight match between flower and pollinator is the product of co-evolution. As the orchid's spur grew longer, it selected for moths with longer tongues. As the moths' tongues grew longer, they selected for orchids with deeper spurs. Each trait drove the other to greater extremes in a self-reinforcing cycle. The result is an almost absurdly precise partnership, two organisms locked in a mutual embrace shaped by millions of years of reciprocal selection. It is one of the most elegant illustrations of co-evolution ever documented, and it began with a quiet prediction by Darwin. Predators and prey are locked in an endless evolutionary arms race. The relationship between predators and their prey is one of the most powerful drivers of natural selection. When a predator evolves a more effective hunting strategy, the prey population comes under strong selective pressure to develop better defenses. Those prey individuals that are slightly faster, more alert, better camouflaged, or more toxic are more likely to survive and pass on their genes. But as the prey population becomes harder to catch, the predator population faces its own selective pressure to become even more effective. This back-and-forth process, known as an evolutionary arms race, can escalate over millions of years. The result is an increasingly sophisticated set of adaptations on both sides. Predators develop sharper senses, stronger jaws, and more cunning hunting behaviors. Prey evolve faster escape responses, more elaborate camouflage, and chemical defenses such as venom or toxins. Neither side ever truly wins. The arms race simply continues, each improvement on one side driving a corresponding improvement on the other. It is a quiet and relentless process, an endless contest that has shaped many of the most remarkable adaptations in the natural world. As cheetahs evolve to run faster, gazelles evolve greater speed too. The cheetah is the fastest land animal on Earth, capable of reaching speeds of over 100 kilometers per hour in short bursts. This extraordinary speed did not arise in a vacuum. It evolved in direct response to the speed of the cheetah's primary prey, the gazelles and antelopes of the African savanna. Over millions of years, natural selection has favored faster cheetahs because those that could not catch their prey did not survive to reproduce. But the same process has also favored faster gazelles because those that could not outrun the cheetah were caught and killed. The result is a finely tuned arms race in which both predator and prey have been pushed to the extreme limits of their physiology. The cheetah has evolved a lightweight build, flexible spine, large nasal passages for rapid oxygen intake, and non-retractable claws for grip. The gazelle has evolved powerful leg muscles, the ability to change direction at high speed, and the stamina to sustain longer chases. Each adaptation in one species is matched by a counter-adaptation in the other. It is a quiet competition played out across the open grasslands generation after generation. This is sometimes called the Red Queen effect. The Red Queen effect takes its name from a character in Lewis Carroll's Through the Looking Glass, who tells Alice that in her country, one must run as fast as one can simply to stay in the same place. In evolutionary biology, the term describes the phenomenon in which species must continually evolve just to maintain their current level of fitness relative to the organisms they interact with. If a prey species becomes faster, the predator must also become faster or face starvation. If a parasite evolves a new way to infect its host, the host must evolve a new defense or face extinction. The Red Queen effect helps explain why sexual reproduction is so widespread. By shuffling genes each generation, sexual reproduction produces genetically diverse offspring, some of which may carry novel combinations of alleles that offer resistance to the latest parasites or predators. Asexual organisms, which produce genetically identical copies of themselves, are more vulnerable to being wiped out by a rapidly evolving enemy. The Red Queen effect is a reminder that evolution is not a race toward perfection. It is a continuous process of relative adjustment, a quiet treadmill on which all living things must keep running simply to survive. Natural selection acts on populations, not individuals. One of the most common misunderstandings about natural selection is the idea that individual organisms evolve. In reality, natural selection acts on populations. An individual organism is born with a particular set of genes and lives its life with those genes largely unchanged. It cannot evolve in response to environmental pressures during its own lifetime. What changes over time is the genetic makeup of the population as a whole. When certain individuals survive and reproduce more successfully than others because of their traits, the alleles that contribute to those traits become more common in the next generation. Over many generations, the average characteristics of the population shift. This is what biologists mean when they speak of evolution by natural selection. The unit of selection is the individual, because individuals differ in their survival and reproduction. But the unit of evolution is the population, because it is the population's gene pool that changes. This distinction is subtle but important. It means that evolution is a statistical process, a gradual shift in the probabilities of different traits across a group. It is a quiet transformation, visible only when viewed across the long arc of generations. Individual organisms do not evolve within their lifetime. An individual giraffe does not grow a longer neck because it stretches to reach high leaves. A single bacterium does not decide to become resistant to an antibiotic. Evolution by natural selection is not something that happens to individuals during their lifetimes. It is something that happens to populations across generations. An organism is born with its particular genetic makeup, and while its body may change in response to its environment through processes like muscle growth or tanning, those changes are not passed on to its offspring. What passes on are genes, and genes do not change in response to an organism's experiences. This was one of the key insights that separated Darwin's theory from earlier ideas, particularly those of Jean-Baptiste Lamarck, who proposed that organisms could inherit acquired characteristics. Natural selection works because some genetic variants happen to produce traits that are better suited to the current environment. Those individuals reproduce more, and their alleles become more common. The individual does not evolve. The population does. It is a slow, patient process, invisible within a single lifetime but unmistakable across many thousands of them. Populations change in allele frequency over generations. At its most fundamental level, evolution is a change in allele frequency within a population over time. An allele is simply a version of a gene, and every population contains a pool of alleles that are shared among its members. When natural selection favors individuals carrying a particular allele, that allele becomes more common in the next generation. When it acts against an allele, that allele becomes rarer. Over many generations, these shifts can be substantial, leading to measurable changes in the traits of the population. Tracking allele frequencies is one of the primary ways that biologists study evolution in real time. In laboratory populations of fruit flies, bacteria, or yeast, researchers can observe allele frequencies changing over just a few dozen generations in response to selective pressures. In the wild, long-term studies of bird populations, insect populations, and plant populations have documented similar shifts. The mathematics of allele frequency change forms the foundation of population genetics, a field that provides the quantitative framework for understanding how natural selection, drift, mutation, and migration interact to shape the genetic composition of populations. Evolution, at its heart, is nothing more than a quiet change in numbers. Genetic drift can also change allele frequencies randomly. Natural selection is not the only force that changes the genetic makeup of a population. Genetic drift, the random fluctuation of allele frequencies from one generation to the next, also plays an important role. Drift occurs because not every individual in a population reproduces, and those that do may not pass on a perfectly representative sample of the population's alleles. In a large population, these random fluctuations tend to average out and have little effect. But in a small population, drift can cause significant changes in allele frequency purely by chance. An allele that is neither beneficial nor harmful may become more common or disappear entirely, not because of its effect on fitness, but simply through the randomness of reproduction. Genetic drift is especially important in newly founded populations, isolated populations, and populations that have recently experienced a dramatic reduction in size. In such cases, drift can cause the population to diverge rapidly from its ancestral gene pool. It is a reminder that evolution is not entirely purposeful. Alongside the orderly process of natural selection, there is always an element of chance, a quiet randomness woven through the fabric of life. In small populations, drift can overpower natural selection. In a large population, natural selection is the dominant force shaping allele frequencies. Beneficial alleles tend to increase and harmful alleles tend to decrease in a predictable manner. But in a small population, the random effects of genetic drift can overwhelm the effects of selection. An allele that is slightly beneficial may be lost by chance, while a slightly harmful allele may become fixed simply because the individuals carrying it happen to reproduce more in a particular generation. This is not because the harmful allele confers an advantage. It is purely a statistical accident, more likely to occur when the number of individuals is small. The smaller the population, the greater the role of drift relative to selection. This is one reason why small, isolated populations are particularly vulnerable to evolutionary problems. They may lose beneficial alleles, accumulate harmful ones, and become less able to adapt to changing conditions. Conservation biologists are keenly aware of this risk and work to maintain population sizes large enough for natural selection to operate effectively. In the quiet interplay between selection and drift, the size of the population often determines which force prevails. Founder effects occur when small groups start new populations. When a small number of individuals from a larger population establish a new, isolated population, they carry with them only a fraction of the original population's genetic diversity. This is known as the founder effect. The allele frequencies in the new population may differ significantly from those in the parent population, simply because the founders were not a representative sample. If the founding group happens to include a high proportion of individuals carrying a rare allele, that allele may become much more common in the new population than it was in the original one. Over time, natural selection and drift continue to shape the new population's genetic composition, but they are working from a different starting point. The founder effect has been documented in many real-world populations. Remote island populations, religious communities that emigrated in small groups, and populations that colonized new habitats after glacial retreat all show the genetic signatures of founder effects. The consequences can be significant, including higher rates of certain genetic conditions that were rare in the ancestral population. It is a quiet reminder that the genetic future of a population can be shaped by the chance composition of its earliest members. The limited gene pool shapes the new population's traits. When a population is founded by a small group of individuals, the genetic diversity available to it is inherently limited. The founders carry only a subset of the alleles present in the original population, and any alleles not represented among them are permanently lost to the new population. This limited gene pool constrains the range of traits that natural selection can act upon in the generations that follow. If the founding group happens to lack alleles for certain beneficial traits, the population may be less able to adapt to future challenges. Conversely, alleles that are common among the founders, even if they were rare in the original population, may come to define the characteristics of the new group. Over many generations, the new population develops its own distinct genetic identity, shaped both by the initial composition of the founders and by the selective pressures of its new environment. This process has been observed in populations ranging from Darwin's finches on remote islands to human communities settled by small migrant groups. The limited gene pool of a founding population is the raw material from which all subsequent diversity must arise. It is a narrow beginning for what may become a rich and varied future. Bottleneck events reduce genetic diversity drastically. A population bottleneck occurs when a large population is suddenly reduced to a very small number of individuals by a catastrophic event, such as a disease outbreak, a natural disaster, or habitat destruction. The survivors carry only a fraction of the genetic diversity that existed in the original population, and many alleles are lost permanently. When the population eventually recovers and grows again, it does so from this reduced genetic base. The result is a population that may be large in number but genetically impoverished. This loss of diversity can have lasting consequences. With fewer alleles to draw upon, the population may be less able to adapt to new diseases, environmental changes, or other challenges. Bottleneck events have been identified in the histories of many species, including elephants, bison, and various species of whale that were hunted nearly to extinction. The genetic signatures of these events can persist for thousands of generations, long after the population has recovered in size. A bottleneck is a sobering reminder that genetic diversity, once lost, cannot be easily regained. It is one of the quiet risks that hangs over every small or threatened population in the natural world. Cheetahs have very low genetic diversity due to a past bottleneck. The cheetah is one of the most striking examples of a species whose genetic diversity has been dramatically reduced by a historical bottleneck. Studies of cheetah DNA have revealed remarkably low levels of genetic variation compared to other large cats. Scientists believe that the cheetah population experienced a severe bottleneck roughly 10 to 12,000 years ago, at the end of the last ice age. During this period, most of the world's cheetah population was wiped out, and the species survived through only a tiny number of individuals. All living cheetahs are descended from this small group of survivors. The consequences are visible today. Cheetahs are so genetically similar to one another that skin grafts between unrelated individuals are not rejected by the immune system, a phenomenon almost unheard of in other species. This extreme genetic uniformity makes cheetahs particularly vulnerable to disease, because a pathogen that can overcome one individual's immune defenses is likely to be effective against all of them. The cheetah situation serves as a powerful illustration of why genetic diversity matters. Speed alone is not enough to ensure survival. Without the quiet variation that fuels natural selection, even the fastest animal on land faces an uncertain future. Mutations provide the raw material for natural selection. Natural selection cannot create new traits. It can only act on the variation that already exists within a population. The ultimate source of all genetic variation is mutation, a change in the DNA sequence of a gene. Mutations arise spontaneously during DNA replication, when the molecular machinery that copies the genetic code makes an error. They can also be caused by external factors such as radiation or certain chemicals. Most mutations affect only a single base pair in the DNA and have very small effects on the organism, if any. But over time, the accumulation of mutations introduces new alleles into the population, providing the raw material upon which natural selection can act. Without mutation, there would be no variation, and without variation, there could be no evolution. This makes mutation one of the most fundamental processes in biology. It is not directed or purposeful. Mutations occur randomly with respect to the needs of the organism. But when a mutation happens to produce a trait that improves survival or reproduction in the current environment, natural selection can amplify it across the population. Every adaptation that exists today began as a quiet accident in a strand of DNA. Most mutations are neutral or harmful, but some are beneficial. The vast majority of mutations have no measurable effect on an organism's fitness. These neutral mutations accumulate silently in the genome over generations, neither helping nor hindering the individuals that carry them. A smaller fraction of mutations are harmful, disrupting important genes or producing proteins that do not function properly. These deleterious mutations are typically removed from the population by natural selection, as the individuals carrying them tend to survive and reproduce less successfully. But very occasionally, a mutation arises that happens to improve an organism's ability to survive or reproduce in its current environment. These beneficial mutations are the ones that natural selection amplifies. A mutation that slightly improves the efficiency of an enzyme, the sharpness of a sense organ, or the resistance to a disease may give its carrier a small but meaningful reproductive advantage. Over many generations, that advantage accumulates and the beneficial allele becomes more common in the population. The ratio of neutral to harmful to beneficial mutations is heavily skewed. Beneficial mutations are exceedingly rare. But because natural selection is so effective at amplifying even the smallest advantages, these rare events are the ones that drive adaptive evolution. It is a quiet lottery, with the occasional winning ticket reshaping an entire species. Beneficial mutations spread through populations over time. When a beneficial mutation arises in a single individual, it initially exists at a very low frequency in the population. But because the mutation improves the carrier's survival or reproduction, the allele is passed on to more offspring than would be expected by chance. In each subsequent generation, individuals carrying the beneficial allele tend to leave more descendants, and the allele becomes progressively more common. This process, known as positive selection, can eventually carry the allele to fixation, meaning that every individual in the population carries it. The speed at which a beneficial allele spreads depends on several factors, including the strength of the selective advantage it confers, the size of the population, and whether other evolutionary forces such as drift or gene flow are also at work. In large populations, even a small selective advantage can lead to the eventual spread of a beneficial allele, though the process may take many thousands of generations. In smaller populations, chance plays a larger role, and even beneficial alleles can be lost through random drift. The spread of a beneficial mutation through a population is one of the most fundamental processes in evolution. It is the quiet mechanism by which adaptation accumulates, one allele at a time. Gene flow between populations can introduce new alleles. Gene flow, also known as migration in a genetic sense, occurs when individuals move from one population to another and contribute their genes to the new population's gene pool. This process can introduce alleles that were previously absent from the receiving population, increasing its genetic diversity. Gene flow is one of the four main mechanisms of evolution, alongside natural selection, genetic drift, and mutation. Its effects can be significant. A single migrant carrying a beneficial allele can introduce that allele to a population that lacked it, potentially providing a new adaptation that improves the population's ability to cope with its environment. Conversely, gene flow can also introduce alleles that are poorly adapted to the local conditions, temporarily reducing the average fitness of the population. The balance between these effects depends on the rate of migration and the strength of local selection. High rates of gene flow tend to homogenize the genetic composition of different populations, making them more similar to one another. Low rates allow populations to diverge independently under the influence of local selection and drift. Gene flow is a quiet thread connecting populations, weaving their genetic fates together across landscapes and sometimes across continents. This can increase genetic diversity within a population. Genetic diversity is the foundation upon which natural selection builds. The more variation a population contains, the more likely it is that some individuals will carry traits suited to new or changing conditions. Gene flow is one of the most important mechanisms for maintaining and increasing genetic diversity within a population. When individuals from a different population arrive and breed with local individuals, they introduce new alleles that may not have arisen by mutation within the local population. This influx of fresh genetic material broadens the range of traits available for natural selection to act upon. Populations that are highly isolated and receive little gene flow tend to have lower genetic diversity and are more vulnerable to the negative effects of inbreeding. Inbreeding increases the likelihood that offspring will inherit two copies of a harmful recessive allele, which can reduce survival and reproduction. Gene flow counteracts this process by introducing new alleles and reducing the frequency of homozygous combinations. In conservation biology, managed gene flow, such as the translocation of individuals between isolated habitat fragments, is sometimes used to rescue populations that are suffering from low genetic diversity. It is a quiet intervention that can have profound effects on the long-term health and adaptability of a population. Stabilizing selection favors intermediate traits. Not all natural selection drives dramatic change. Stabilizing selection is a mode of selection that favors the average or intermediate form of a trait, acting against individuals at either extreme. When a trait is already well suited to the current environment, natural selection tends to remove the outliers and maintain the existing optimum. This has the effect of reducing variation within the population and keeping the trait distribution narrow and centered around the mean. Stabilizing selection is thought to be the most common mode of natural selection in nature because most populations are already reasonably well adapted to their environments. It is the reason why many traits in established populations remain remarkably consistent from generation to generation. The selective removal of extreme variants prevents the population from drifting too far from the traits that have proven successful in the past. Stabilizing selection does not produce the dramatic evolutionary changes that capture public attention. It works quietly in the background, maintaining the status quo and preserving adaptations that have been refined over countless generations. It is the conservative force in evolution, the gentle pressure that says not everything needs to change. Sometimes, staying the same is the best strategy. Human birth weight is under stabilizing selection. One of the most well-documented examples of stabilizing selection in humans involves birth weight. Studies conducted over many decades have consistently shown that babies of intermediate birth weight have the highest survival rates. Babies that are significantly lighter than average are more vulnerable to infection, have less energy reserves, and may struggle to regulate their body temperature. Babies that are significantly heavier than average face increased risks during delivery, including complications for both the infant and the mother. As a result, natural selection has historically favored babies whose weight falls within a moderate range, neither too light nor too heavy. This is a classic example of stabilizing selection acting on a continuous trait in a human population. The optimum birth weight reflects a balance between the costs and benefits of larger and smaller body size at birth. In modern medicine, advances in neonatal care and surgical delivery have reduced the mortality associated with extreme birth weights, potentially relaxing the selective pressure. But for most of human evolutionary history, this stabilizing force was a significant influence on the distribution of birth weight. It is a quiet example of natural selection operating within our own species, shaping one of the most fundamental aspects of human biology. Babies that are too small or too large face higher risks. The relationship between birth weight and survival is a clear illustration of how natural selection penalizes extremes when the optimum lies in the middle. Newborns who weigh significantly less than average are at greater risk of hypothermia because they have a higher surface area to volume ratio and lose heat more rapidly. They are also more susceptible to infections because their immune systems may be less developed. At the other end of the spectrum, unusually large babies face increased risks during labor and delivery. For most of human history, before the advent of modern surgical techniques, a very large baby could pose a serious threat to the survival of both the infant and the mother. These opposing risks create a selective pressure that favors babies of moderate weight, resulting in the stabilizing selection pattern observed in population studies. The distribution of birth weights in human populations tends to cluster around the optimum, with relatively few individuals at the extremes. This pattern has been remarkably consistent across diverse populations and historical periods. It is one of the most accessible examples of natural selection acting in a measurable way on a human trait. The quiet pressure of stabilizing selection has shaped this distribution over thousands of generations, long before modern medicine began to soften its edges. Directional selection shifts traits toward one extreme. Directional selection occurs when individuals at one end of a trait distribution have a fitness advantage over those at the other end. Over time, the average value of the trait in the population shifts in the favored direction. This mode of selection is most commonly observed when environmental conditions change, creating new selective pressures that favor traits that were previously neutral or even disadvantageous. For example, if a prolonged drought reduces the availability of soft seeds and leaves only hard, thick-shelled seeds, birds with larger, stronger bills will have an advantage. Over several generations, the average bill size in the population will increase as natural selection favors the larger-billed individuals. Directional selection is the mode most readily associated with adaptive evolution because it produces measurable change in a trait over time. It has been documented in many natural populations, including the famous studies of beak size in Darwin's finches on the Galapagos Islands during periods of drought. The strength and speed of directional selection depend on the magnitude of the environmental change, the heritability of the trait, and the amount of standing genetic variation in the population. It is a quiet but relentless force, gradually reshaping populations in response to the shifting demands of their environment. This occurs when environments change consistently. Directional selection is most apparent when the environment shifts in a consistent direction over an extended period. A gradual warming of the climate, a sustained increase in the prevalence of a particular predator, or a long-term change in the availability of a key food source can all create conditions under which one extreme of a trait distribution is consistently favored over the other. The organisms best suited to the new conditions survive and reproduce at higher rates, while those less suited decline. Over many generations, the population shifts toward the favored extreme. This is how natural selection drives adaptation, not through sudden leaps, but through the slow accumulation of small advantages over time. Environments rarely remain static. Seasons shift, climates change, new competitors arrive, and diseases emerge. Each of these changes can trigger a new episode of directional selection. In some cases, the direction of selection may reverse entirely if conditions change again. A trait that was advantageous in one era may become a liability in the next. Directional selection is therefore not a one-time event, but a recurring process, continuously adjusting the characteristics of populations to match the conditions they face. It is a quiet recalibration, happening so slowly that it is often invisible within a single human lifetime. Disruptive selection favors both extremes over the middle. Disruptive selection is the opposite of stabilizing selection. Instead of favoring the average, it favors individuals at both extremes of a trait distribution while selecting against those in the middle. This creates a bimodal distribution, with two peaks at either end and a valley in the center. Disruptive selection can occur when a population occupies an environment with two distinct niches, each favoring a different set of traits. For example, in a habitat where food sources include very large seeds and very small seeds, but few medium-sized ones, birds with either large or small bills may be favored, while those with intermediate bills are at a disadvantage. Disruptive selection is thought to be less common than directional or stabilizing selection, but it has been documented in several natural populations. It is of particular interest to evolutionary biologists because it has the potential to increase variation within a population rather than reduce it. By maintaining two distinct forms, disruptive selection can create the conditions for the population to split into two separate groups, each adapted to its own niche. It is one of the mechanisms by which a single population can begin the journey toward becoming two species, a quiet fork in the road of evolution. This can lead to the formation of new species. Disruptive selection, by favoring two different extremes of a trait and selecting against intermediates, can gradually divide a population into two distinct groups. If the two groups begin to mate preferentially with individuals that share their own trait values, gene flow between them decreases. Over time, the groups may diverge genetically and phenotypically to the point where they can no longer interbreed. At that stage, they are effectively two separate species. This process, known as sympatric speciation, does not require a physical barrier to separate the populations. The selective pressures themselves create the divide. Sympatric speciation is more controversial among biologists than allopatric speciation, and there is ongoing debate about how common it is in nature. However, there are several well-studied examples that suggest it does occur under the right conditions. Cichlid fish in African lakes, apple maggot flies in North America, and certain plant species that have diverged in pollination strategy are all cited as possible cases. Disruptive selection provides the initial push, but assortative mating and reinforcement of reproductive barriers are needed to complete the process. It is a quiet pathway to speciation, requiring no mountains or oceans, only the persistent pressure of natural selection acting in two directions at once. Frequency-dependent selection favors rare traits. Frequency-dependent selection is a form of natural selection in which the fitness of a trait depends on how common or rare it is in the population. In negative frequency-dependent selection, rare traits have an advantage precisely because they are rare. Predators, for example, tend to form a search image for the most common prey type, which means that individuals with unusual appearances are less likely to be detected and captured. As a rare trait becomes more common, its advantage diminishes and other rare variants gain the upper hand. This creates a dynamic cycle in which no single form ever dominates the population permanently. The result is the maintenance of multiple forms within the population, a phenomenon known as polymorphism. Negative frequency-dependent selection is one of the mechanisms by which genetic diversity is maintained over long periods. It has been observed in many species, from fish and lizards to plants and bacteria. The key insight is that rarity itself can be an advantage in a world where common traits attract disproportionate attention from predators, parasites, or competitors. It is a subtle and elegant form of natural selection, a quiet balancing act that rewards those who dare to be different. Rare color morphs in guppies may avoid predators more easily. Guppies are small freshwater fish that display a remarkable variety of color patterns, particularly among males. Research conducted in streams in Trinidad has shown that male guppies with rare color patterns tend to survive longer than those with common patterns. The reason is straightforward. Predators that feed on guppies form a mental search image based on the most frequently encountered color pattern. Males whose coloration differs from the majority are less likely to match the predator's search image and therefore less likely to be caught. This gives rare color morphs a survival advantage, an example of negative frequency-dependent selection in action. As a rare pattern becomes more common through the reproductive success of its carriers, predators begin to recognize it, and the advantage fades. Meanwhile, other previously common patterns become rarer and gain the advantage in turn. This cycling of color morph frequencies maintains a high level of visual diversity within the guppy population. The guppy system has become one of the best-studied examples of frequency-dependent selection in the wild. It demonstrates that being different can sometimes be the best strategy for survival. In the quiet currents of a tropical stream, rarity is its own reward. Balancing selection maintains multiple alleles in a population. Balancing selection is a broad term for any form of natural selection that maintains genetic diversity within a population by favoring the persistence of more than one allele at a given gene. It encompasses several mechanisms, including heterozygote advantage, negative frequency-dependent selection, and selection that varies across time or space. The common thread is that no single allele is driven to fixation or elimination. Instead, multiple alleles coexist in a stable equilibrium. Balancing selection is important because it preserves the genetic variation that populations need to respond to future environmental changes. Without it, natural selection would tend to drive allele frequencies to extremes, reducing diversity over time. Balancing selection counteracts this tendency, maintaining a reservoir of alleles that may prove useful under different conditions. Some of the best-known examples of balancing selection involve disease resistance. The major histocompatibility complex, which plays a central role in the immune system, is one of the most genetically diverse regions of the human genome, and balancing selection is thought to be a major reason why. By maintaining diversity at key genes, balancing selection helps populations cope with the ever-changing landscape of infectious disease. It is a quiet guardian of variation, ensuring that the genetic toolkit of a population remains well-stocked for whatever challenges lie ahead. The sickle cell allele is maintained because heterozygotes resist malaria. One of the most famous examples of balancing selection involves the sickle cell allele in human populations. Individuals who inherit 2 copies of the sickle cell allele develop sickle cell disease, a serious condition in which red blood cells become misshapen and can block blood vessels, causing pain and organ damage. Individuals who inherit 2 copies of the normal allele have healthy red blood cells but are fully susceptible to malaria. However, individuals who inherit 1 copy of each allele, the heterozygotes, enjoy a remarkable advantage. Their red blood cells are resistant to the malaria parasite, and they do not suffer the severe symptoms of sickle cell disease. In regions where malaria is prevalent, heterozygotes have a significant survival advantage over both homozygous types. This is known as heterozygote advantage, and it is a powerful form of balancing selection. It maintains both alleles in the population at stable frequencies because neither allele can be driven to fixation without eliminating the fitness advantage of the heterozygote. The sickle cell example illustrates a central truth about natural selection. It does not produce perfect solutions. It produces trade-offs, quiet compromises between competing pressures, shaped by the specific conditions of each environment. Artificial selection is natural selection guided by humans. Artificial selection is the process by which humans deliberately choose which individuals in a population are allowed to breed based on desired traits. The underlying mechanism is identical to natural selection. Individuals with certain heritable characteristics are more likely to reproduce and pass on their genes. The difference is that the selective agent is a human breeder rather than the natural environment. Artificial selection has produced an astonishing range of domestic plants and animals, from the hundreds of dog breeds that exist today to the high-yielding crop varieties that feed billions of people. In each case, humans identified traits they valued, such as size, speed, coat color, milk production, or grain yield, and preferentially bred the individuals that displayed those traits. Over many generations, the cumulative effect of this selective breeding has produced organisms that differ dramatically from their wild ancestors. Artificial selection demonstrates the power of selection to reshape organisms over relatively short periods. What took natural selection millions of years to achieve, directed human selection can accomplish in hundreds or thousands of generations. It is a quiet testament to the principle that variation plus selective pressure equals change, whether the pressure comes from nature or from the deliberate hand of a breeder. All domestic dogs descended from wolves through artificial selection. Every domestic dog on Earth, from the smallest Chihuahua to the largest Great Dane, is descended from the gray wolf. The domestication of dogs is thought to have begun somewhere between 15,000 and 40,000 years ago, making the dog the first domesticated animal. The process likely started when wolves that were less fearful of humans began scavenging near human camps. Those that were most tolerant of human presence were gradually accepted and eventually bred selectively for traits that humans found useful, such as herding ability, guarding behavior, hunting skill, or simply companionship. Over thousands of generations, artificial selection transformed the wolf's descendants into the extraordinary diversity of breeds we see today. Some breeds have been selected for speed, others for strength, others for a keen sense of smell, and still others for their appearance or temperament. The genetic differences between breeds can be substantial, yet all dogs remain a single species capable of interbreeding. The story of the domestic dog is one of the most vivid illustrations of how powerful selective breeding can be. Starting from a single wild ancestor, human selection has produced a range of forms that rivals the diversity seen in entire families of wild animals. It is a quiet revolution, achieved not in laboratories, but in kennels and farmyards across the centuries. Crops like maize were shaped from wild grasses over thousands of years. Modern maize, one of the most important food crops in the world, is almost unrecognizable compared to its wild ancestor, a small grass called teosinte that still grows in parts of Mexico. Teosinte produces tiny ears with just a handful of hard, inedible kernels enclosed in a tough casing. Through thousands of years of selective breeding by ancient farmers in Mesoamerica, teosinte was gradually transformed into the large, soft-kerneled maize that we know today. Each generation, farmers chose the plants with the largest ears, the most kernels, and the softest casings, and used their seeds for the next planting. This process of artificial selection reshaped the plant's architecture, kernel size, and nutritional content to an extraordinary degree. The transformation from teosinte to maize is one of the most dramatic examples of artificial selection in the history of agriculture. It demonstrates that the same principles underlying natural selection, heritable variation, and differential reproduction can produce profound changes when directed by human intent. Similar stories can be told for wheat, rice, and many other staple crops that were shaped from wild grasses by generations of patient human selection. Every grain of maize is a quiet monument to the power of cumulative selective change. Darwin studied artificial selection to understand natural processes. Charles Darwin devoted considerable attention to artificial selection, recognizing that the practices of breeders could illuminate the mechanisms of natural selection. In the opening chapters of On the Origin of Species, he described in detail how pigeon fanciers, cattle breeders, and horticulturists had produced astonishing variety among domesticated organisms by consistently selecting for desired traits over many generations. Darwin argued that if human breeders could achieve such dramatic transformations in a few hundred years, then nature, operating over millions of years and across entire continents, could produce changes of even greater magnitude. Artificial selection provided Darwin with a familiar, observable analogy for a process that was far too slow to witness directly in the wild. By studying the techniques and results of selective breeding, Darwin was able to establish several key principles. He showed that variation exists within populations, that much of this variation is heritable, and that selective breeding can shift the characteristics of a population in a predictable direction. These principles applied equally well to nature, where the environment itself acts as the selective agent. Darwin's use of artificial selection as a bridge to understanding natural selection was one of the most effective rhetorical strategies in the history of science. It turned the quiet observations of farmers and breeders into evidence for a grand unifying theory of life. He corresponded with pigeon breeders to learn about variation. Darwin was a passionate pigeon breeder himself and maintained a large collection of domestic pigeon varieties at his home in Kent. He corresponded extensively with pigeon fanciers across Britain and Europe, seeking detailed information about how breeders selected for specific traits such as plumage color, body shape, beak length, and tail feather arrangement. The diversity among domestic pigeon breeds astonished him. Some had enormous fan-shaped tails, others had inflatable throat pouches, and others had feathered feet or distinctive crests. Yet all of them descended from a single wild species, the rock dove. Darwin realized that this diversity had been produced by generations of breeders choosing which birds to mate, a direct parallel to the way natural selection acts on wild populations. His correspondence with breeders provided him with detailed case studies of how new varieties arise, how traits are inherited, and how rapidly change can occur under consistent selective pressure. These practical observations gave Darwin confidence that the same process, operating over far longer timescales, could produce the diversity of life on Earth. The pigeons of Victorian England, bred in lofts and shown at competitions, became quiet witnesses to one of the greatest scientific insights of all time. Natural selection explains the complexity of the eye. The eye has long been cited as a challenge to the theory of evolution by natural selection. How could such a complex organ, with its precisely curved lens, its light-sensitive retina, and its intricate neural connections, have arisen through gradual, undirected change? Darwin himself acknowledged the apparent difficulty, but argued that even a slight ability to detect light would confer a survival advantage. From that starting point, natural selection could refine and elaborate the visual system in small, incremental steps over millions of years. Modern evolutionary biology has confirmed this reasoning. Studies of living organisms reveal a complete range of eye types, from simple clusters of light-sensitive cells in flatworms to the sophisticated camera eyes of vertebrates and cephalopods. Each intermediate stage is functional and provides a clear advantage to its possessor. A simple eye cup that can detect the direction of light is better than no eye at all. A pinhole eye that can form a rough image is better than an eye cup. A lens that focuses light sharply is better than a pinhole. Natural selection does not need to produce the finished product in a single step. It only needs each intermediate to be slightly better than the one before. The complexity of the eye is not an argument against evolution. It is one of its finest demonstrations. Simple light-sensitive cells evolved into complex camera eyes. The evolution of the eye can be traced through a series of stages that are still represented by living organisms today. The simplest light-detecting structures are individual photoreceptor cells, found in some single-celled organisms and in the simplest multicellular animals. These cells can detect the presence or absence of light, but cannot form an image. The next stage is a cluster of photoreceptor cells arranged in a shallow depression or eye cup, which allows the organism to detect the direction of light. Flatworms possess this type of eye. A deeper cup creates a crude pinhole camera effect, enabling rough image formation. The nautilus, a living relative of the ammonites, has an eye of this type. The addition of a transparent covering protects the eye and can act as a primitive lens. Further refinement of the lens produces the sharp focusing camera eyes found in vertebrates and cephalopods, such as octopuses and squid. Each of these stages is functional and provides a meaningful survival advantage in its own right. Computer simulations have estimated that the transition from a flat patch of photoreceptors to a fully focused camera eye could occur in fewer than 500,000 years, a relatively short span in evolutionary terms. The eye's evolution is not a mystery. It is a quiet triumph of incremental improvement, driven by the relentless advantage of seeing just a little bit better. Each small improvement offered a survival advantage. The key to understanding the evolution of complex structures like the eye is the principle that each small improvement along the way offered a real and measurable advantage to the organism that possessed it. An organism with a simple patch of light-sensitive cells can detect shadows, which helps it avoid predators. An organism with a slightly curved eye cup can determine the direction of light, which helps it orient toward or away from threats. An organism with a primitive lens can form a rough image, which allows it to identify objects in its environment. At every stage, the individual with the slightly better visual system is more likely to survive and reproduce than one without it. This cumulative advantage drives the progressive refinement of the structure over many generations. No single mutation needs to produce a dramatic improvement. The advantage of each step can be very small as long as it is consistent. Over thousands and millions of generations, these small advantages compound into the extraordinary complexity we observe today. This principle applies not only to the eye, but to every complex adaptation in the biological world. Wings, immune systems, echolocation, and photosynthesis all evolved through the same patient process of incremental refinement. It is a quiet accumulation of tiny gains, each one building on the last. The fossil record documents evolution through natural selection. The fossil record provides some of the most compelling evidence for evolution by natural selection. Fossils are the preserved remains or traces of organisms that lived in the past, embedded in layers of sedimentary rock. By studying the sequence of fossils across geological time, paleontologists can trace the gradual changes in body form, size, and complexity that populations have undergone over millions of years. The oldest rocks contain the simplest organisms, single-celled life forms that dominated the Earth for billions of years. As we move to younger and younger rock layers, we see the appearance of increasingly complex organisms, from multicellular animals to fish, amphibians, reptiles, mammals, and eventually the diverse ecosystems of the modern world. The fossil record is not complete. Fossilization is a rare event, and many organisms have left no trace. But where fossils are abundant, they reveal clear patterns of change that are fully consistent with the predictions of evolutionary theory. Species appear, persist for a time, change, and eventually go extinct, replaced by new forms better suited to the conditions of their era. The fossil record is a quiet archive of life's long history, written in stone and waiting to be read by those who know how to look. Transitional fossils show intermediate forms between species. Transitional fossils are among the most powerful pieces of evidence for evolution. They are fossils that display features intermediate between two groups, showing how one form gradually transformed into another over time. For many years, critics of evolution pointed to apparent gaps in the fossil record as evidence against the theory. But as paleontology has advanced, many of those gaps have been filled by remarkable transitional fossils. Archaeopteryx, discovered in the 19th century, has the feathered wings of a bird but the teeth, clawed fingers, and bony tail of a reptile. Tiktaalik, found in the Canadian Arctic, has the body of a fish but the limb structure and flat head of an early land animal. Ambulocetus, an early whale ancestor, had legs capable of walking on land and a body adapted for swimming. Each of these fossils captures a moment in an evolutionary transition, a snapshot of a lineage in the process of becoming something new. Transitional fossils do not represent halfway stages. Every organism is fully adapted to its own way of life, but they demonstrate that the differences between major groups of organisms did not appear all at once. They accumulated gradually, driven by natural selection operating step by step. It is a quiet testimony, etched in the rock, to the power of incremental change. Tiktaalik is a transitional fossil between fish and land animals. Tiktaalik rosie is one of the most celebrated transitional fossils ever discovered. Found in Arctic Canada, In rocks dating to roughly 375 million years ago, Tiktaalik lived during the late Devonian period, a time when vertebrate life was beginning the transition from water to land. Its body displays a remarkable combination of fish and tetrapod features. Like a fish, it had scales and fins. But like an early land animal, it had a flat, crocodile-like head, a flexible neck, and robust fins supported by bones homologous to the wrist and finger bones of later tetrapods. These sturdy fins could have been used to prop the body up in shallow water or to push along muddy substrates. Tiktaalik is not considered a direct ancestor of all land vertebrates, but it represents the type of organism that bridged the gap between fish and the earliest four-legged animals. Its discovery was particularly remarkable because the research team predicted where to look based on the age and type of rock formations likely to contain such a fossil. This combination of prediction and discovery is a powerful demonstration of evolutionary theory's explanatory power. Tiktaalik is a quiet ambassador from one of the most pivotal transitions in the history of life. Natural selection continues to shape life on Earth today. Natural selection is not a process confined to the distant past. It is happening right now in every ecosystem on the planet. Bacteria are evolving resistance to antibiotics in hospitals and farms. Insects are adapting to pesticides in agricultural fields. Fish are evolving smaller body sizes in response to the selective pressures of commercial fishing, which preferentially removes the largest individuals. Urban environments are creating new selective pressures on birds, mammals, and plants that must adapt to noise, light pollution, and fragmented habitats. Even within human populations, natural selection continues to operate, albeit in ways that are complicated by modern medicine and technology. The emergence of new infectious diseases, the spread of existing ones, and the changing global climate are all creating new selective pressures that will shape the future of life on Earth. Evolution by natural selection is not a completed chapter. It is an ongoing story, written in real time by every organism that lives, reproduces, and dies on this planet. As you drift towards sleep tonight, somewhere in the world a mutation is arising, a selective advantage is being tested, and a population is quietly changing. The process that produced all the diversity of life continues, unhurried and relentless, into the future. And now, the quiet wonders of natural selection begin to gently fade into the stillness of the night. You have wandered through over 100 peaceful truths about evolution's patient engine. From Darwin's voyage on the Beagle to the antibiotic resistance unfolding in hospitals today. From the peppered moth's shifting colors to the peacock's extravagant tail. From the finches of the Galapagos to the silent mutations reshaping genomes every moment. You have explored how variation arises and how selection sifts through it, how predators and prey shape each other across millennia, and how isolated populations diverge into new species. You have learned that natural selection has no plan or purpose, only the simple truth that some variations survive better than others. And yet, from that simplicity has emerged all the astonishing complexity of life on Earth. You are connected to this story more deeply than you may realize. Right now, as you listen, natural selection continues its quiet work in ecosystems around the world. Bacteria evolve in hospital wards, insects adapt to changing climates, and somewhere, a mutation is arising that may one day shape a future species. If your eyes feel heavy now, let them close. Let the rhythm of your breath become steady, like the slow, patient accumulation of small changes across deep time, like the gradual transformation of populations over countless generations. Tonight, as you rest, evolution continues its ancient work all around you. Species adapt and diverge, genes flow between populations, and the tree of life grows ever more intricate. You are a living branch on that tree, the latest expression of nearly 4 billion years of unbroken ancestry. You are a warm presence within a world shaped by patient, relentless change, peacefully resting among the wonders of life's long story. Sleep well, sweet dreams, and may you carry with you the quiet knowledge that you are part of something far older and grander than any single lifetime. That every feature of your body, every instinct you carry, was shaped by the same process that produced all life on Earth. That somewhere in this very moment, natural selection is still at work, sculpting the future from the raw material of variation. Thank you for joining me on this journey through the remarkable world of natural selection. If you enjoyed this video, please subscribe to Drowsy Discovery for more soothing explorations of the wonders hidden in our world. There are more wonders waiting, more secrets hidden in the history of life on Earth, more gentle truths to drift through on quiet evenings. Rest well, and may the quiet patience of evolution keep you company through the night. This has been Drowsy Discovery. Good night.